Okay, good morning and welcome to the session on bioremediation. My name is Alan Seach. I'm here from California. I'm with Evonik Corporation, and I'd like to introduce our first four speakers. Number one is Dr. Danny de Graaff from TAU, who works on soil and groundwater remediation. Please come up, Danny. Our second speaker is Cosimo Masini, who's also co-chairing the session. Third is David Aguilar, and the fourth speaker is Jesica Soder. We have an interesting session filled with 13 presentations on bioremediation, with a lot of focus on lindane and other HCH compounds. The presentations cover a broad range of science and technology, microbiology, chemistry, biochemistry, both aerobic and anaerobic bioremediation, and both soil and groundwater. So I hope you'll enjoy the interesting session.
Well, my name is Danny de Graaff. I'm from TAU, a soil and groundwater consultant, and I'm going to present a bit about microbiome-based remediation and other nature-based techniques. So this presentation was also, the abstract was also written by my colleague Tobias Praamstra and by Cosimo Masini, who is chairing the session today. So the overview of what I'm going to present. First, I'm going a bit into the scope and context about the remediation challenges we have. Then I'm going to dive a bit more into the EU projects that are focused around nature-based solutions. And then I'm going to talk a bit about two current projects that are running. So first, the Bioremediation project. It's a new project that started just half a year ago, and the other one is a natural catch solution that has been running for the past 4.5 years. So first, the scope and context. Well, there's now the EU soil strategy, in which they say that, they actually say themselves that there's a massive soil and groundwater remediation task in EU countries, which is already a big step if the EU says it, so we can really have a good start on remediating that. And they say that about 14% of 2.8 million potentially polluted sites are expected to require remediation. So there's 390,000 sites, which is quite a big challenge, I'd say. And out of that, there was, by 2018, there were only 65,000 remaining, but still a pretty large part. And then, specifically about HCH, about 300 sites were, it was handled in EU countries. And yeah, the major part still needs to be remediated, but I don't think I need to tell this to this audience. Well, an important part about the whole remediation thing is that you shouldn't just only think about the contamination itself, but it's part of a bigger structure. So you need to think about the environmental, the social, and the economic value. And an important part about that is the nature-based solutions. So instead of just digging away and just using massive polluting techniques, we want to look into what are more nature-based solutions that are more sustainable, they're cheaper, less disturbing, they emit less CO2, and they have also, very important part, less extensive maintenance. So you can make it run for longer without actually having to go into the field. So these are some examples of NBS processes and mechanisms. So we can make use of certain aspects that are found in nature. Well, the first quite easy one is the natural flow rate of groundwater as a driving force. I mean, it's flowing, just use it. A second one is capping with natural minerals. Also the use of green adsorbents and minerals for adsorption. Then an important one is the use of bacteria and fungi, because they're known to be able to degrade certain compounds. Another one is, limestone gravel beds. They can decrease pH and increase immobilization, in which you can stabilize your pollution. And another interesting one is phytoremediation, which you can use trees and vegetation for capturing your pollution. So there are a few EU projects. that are nature-based, and they're listed quite right here. And the most important thing is that they are, well, based on nature-based solutions. So the first one is about bioelectrochemical techniques, and it's also used for pesticides, which is quite an important link for this forum, of course, for HCH. And then for Regenat, there was a remediation pilot with phytoremediation, in which you can also have some more oil-based compounds. The live pulp wat, who's also present here, is also using constructed wetlands for removing pesticides in HCH contaminated waters. Then there's the H2020 recycle, which is also involved in pesticide removal. There's the H2020 remedy, which is also using some water nature-based solutions. And then the other one that I'll present in a few minutes is the Horizon Europe MIBREM project, in which we're looking into microbiome-based remediation of HCH, PAH, and cyanide. Okay, so now we'll go into two projects that are currently running. The first one I want to mention is the MIBREM. The title is an innovative toolbox for microbiome-based soil remediation. It's an EU Horizon project that has been started only half a year ago in October 2022, and it will run for a total of 4.5 years. And we want to look into, well, bioremediation of HCH for nature-based remediation. So we have 11 partners based over 6 countries in Europe. Here's the project consortium. There's a lot of different partners, and you can see a picture of us. We just had our kickoff meeting last year in Vienna. So it's a mix of different universities, different companies, research institutes, and together we want to make the whole look into the HCH remediation. So the goal and the challenge of the MIBREM project is, well, it's written here. It will leverage the potential of microbiomes for bioremediation of contaminated sites, but developing unique and innovative technology toolbox. And to go a bit more into detail is how we want to do that. We want to identify, by using and developing molecular methods, isolate, cultivate, and upscale microbiomes for bioremediation to pilot scale, for sites contaminated with, among others, HCH. So we want to go into the sites. We know we have already established which sites are currently contaminated with HCH. We want to go to those sites, take the samples. Then our partners at universities want to characterize what's in there, which type of bacteria, also which kind of genes are responsible for degrading HCH. What are the optimal conditions for growth for them? Which kind of nutrients do they need? And then we want to go back to the field and use a pilot scale study in which you can actually use these bacteria and the knowledge that we have, and then implement it in a pilot scale study to actually remediate the HCH. So so that's the last part that's on the slide. It's the long-term upscaling in the end of microbiome-based remediation. So after the pilot, we want to keep on running with these techniques. So in order to have this big task completed, we divided it up in a few work packages. Well, work package 1 is the one that we're currently working on. It's the quest for contaminated sites. So we have a big list already. Well, we have a list of the sites. We want to now go into the field for sampling, taking the samples in order to get it to the university partners. And then we're going to the next work packages, microbiome analysis and microbiome and strain collection, which are focused at the university. So they're really going into microbiology and into metagenomics, into transcriptomics, seeing what they can do with these bacteria. Then work package 4 is looking into modeling as well. If you know beforehand what you can expect, it's easier than actually always measuring. So the building of a modeling tools is one part. And then work package 5, we want to go back into the field, do the upscaling and field testing. Then the last 2 work packages are also quite important. First is the regulation and business development, because we can do, from a more scientific point of view and engineering point of view, we can do a lot, but if it's not regulated or if there's no business plan, you can forget about it in the long run. And the other one is important is sharing results and communication. As you all might know, I mean, it's quite a sensitive topic, especially to people living around it, why we want to do this and how we can actually achieve a better environment. So we just don't want to only focus on, you know, the technology and the boring numbers, but it's part of a bigger plan. So we really want to make sure that we are capturing that aspect of the project as well. So currently we have pre-selected some potential HCH sites. They're listed here. We have currently, there's 6 listed here. There's the first one that's also in cooperation with Live Pop Wat. So we can go into their Polish sites that they're starting up right now. And there's a few in France, Germany and Spain that we want to use. Well, currently we're contacting the owners and planning the field work to actually get it started. Okay, then I'll quickly go into the natural catch. This was a project that has been running for the past years. The situation is, it was based in Ghent in Belgium. It was a cooperation between Netherlands and Flanders. It was a former tar and black factory site, carbon black factory site, with a large residual contamination. Well, you can see the influx. It's mainly hydrocarbons and some benzene, naphthalene, and some other nasty compounds. And they have a very high concentration in the water. And the challenge there was to have a structural improvement of the water quality. And the demand is also, we want to have a nature-based solution. And the solution that was come up with is the reactive meth, or the natural catch, which uses 3 pillars of nature-based solutions. So first, they use the natural drainage capacity of the canal. Second, the use of a green adsorbent in the mat. And then a biologically active interface on the mat for active biodegradation. So you first capture, in your mat, you capture your contaminant, and then some bacteria can actually degrade it in a natural way. So pictures are always the best way to show it. So here are a few. So you can see that there are, well, it's along the whole river, there are mats, parts, which are covered with adsorbent. So part of it is by biochar, part with peat, and they can actually capture the contaminant, and subsequently bacteria can remove it biologically. So with that, we have reduced PAH, BTEX, and C6, C10 oil compounds with, well, close to 100%, and benzene a bit less because it's more mobile, so it takes, it's a bit more challenging. And by that, we, well, it's a good start for future developments as well. And the bottom one, it says also, well, for pesticides, LINDANE, HCH, it can also be used by similar kind of absorption mechanisms, and afterwards you can have some biodegradation occurring on the reactive mat. Okay, so I want to finish with these takeaway messages. So first, well, nature-based solutions are key regarding the number and complexity of sites in the EU, and it's also what the EU itself is really pushing. And yeah, like I discussed, there's a few EU projects that have been initiated, like the REMEDIATION and the MICROBIOME project. And finally, NBS can be feasible for HCH site because of the cost effectiveness and the applicability on large remote sites. Thank you.
Good morning, everybody. Thanks, Danny. Thanks, Alan. My presentation will be in line with what already presented by Danny, and I'm going to focus in particular on the sustainable approach to the biodegradation of LINDANE. So what's on the agenda? I'm going to briefly present D&D Biotech, and then I'm going to touch quickly on the history of LINDANE and the HCH isomers, and then dive into a case study with the activities done so far, the results, and the next steps. And then finishing with the presentation of our pilot system that we'll be using also for this work. D&D Biotech is an Italian company based in Pisa, but we mainly work in Europe, South America, and Middle East. We are a multidisciplinary team, and I think this is quite important due to the complexity of these subjects, such as LINDANE and HCH. I like to say that our work starts from micro and goes to macro, in the sense that we first study the microbial ecology of soil and groundwater in order to understand the microorganisms that are present in contaminated matrices and isolate them, reproduce them, and inoculate them back in order to accelerate natural occurring degradation processes. So to do this, we apply molecular techniques, metagenomics, we do lab testing, we do pilot testing, prototyping, and ultimately we also produce the autochthonous biomass for the bioaugmentation. And on top of this, we have developed a technique related to the use and modification of natural zeolites as a filtering media for mainly heavy metal removals. Having said that, let's move on to LINDANE. Obviously, I mean, it's a focus of the forum, so you certainly know a lot about it, but just a quick recap in terms of use that's been going on for more than 50 years. And as we know, the production process was very inefficient. 1:8, 1:12 ratio, LINDANE produced, byproducts discharged. So this gives an idea of the extent of the waste that were produced, and obviously without any proper legislation in place in that period, which resulted in illegal disposal and production facilities, which resulted in very numerous and big uncontrolled landfills. A total of almost 5 million tons of HCH waste is present worldwide, and as we know, obviously being a POP, it's barely degrades in the environment, it bioaccumulates, and it presents all sorts of different problems along the food chain and risk to human health. So in short, it was banned, the production, and then it was inserted in the Stockholm Convention in 2009. Our work focuses on the possibility of biodegrading these molecules. So our focusing really is to exploit these routes, whether it's bacterial degradation, whether it's fungal degradation, aerobic, anaerobic. But our objective is really to tackle the diffuse contamination. And as we will see from our case study, which I can actually present now, there are two main problems. There are the industrial areas where there is the source, primary or secondary contamination. So it's relatively small, but volumes with high concentrations. And then there is the diffuse contamination. So in the case of this site in central Italy, which is called Valle del Sacco, the region, the industrial area is named Colleferro, and the production took place from mid-40s to the late 70s, with resulting disposal area in the industrial part. So as you can see, the red dot represents the industrial area, which is Colleferro, and then you have the river Sacco, which gives a name to the valley, and unfortunately there were several transports and mechanisms and diffusions, aerial, water, groundwater, that resulted also in the contamination of the sediments of the river. And we are talking about an area of around 5,500 hectares and about 60, 70 kilometers of spread along the river. Sediments, water, and soil. The problem came out in 2005 when high concentrations of isomers were detected in cow milk in the area, because it's an area with various farms. And after a detailed study, basically farming and grazing was banned in the area, and it is still banned now, because the focus, as it makes sense, at the beginning has been the industrial area, so removal of the primary sources of contamination. and the budget available for the public authorities was allocated for these main activities, resulting in the construction of hazardous landfill in the site for the removal of approximately 70 to 80,000 cubic meters. But obviously the problem, as I mentioned, is not only there, it's diffused. So the next steps, as we already started to do also in the HCH in EU project that will be presented later on, is focusing on the remaining diffuse contamination, in particular in river and agricultural areas. And our path, which also constitutes a bit of a background to the MIBIREM project that was presented by Danny, was to focus on the biodegradation. This is just an image of the industrial area with white substance findings. We're talking about 2,500 cubic meters of white soil, let's say, where about 50% was constituted by HCH isomers, and 1,000 cubic meters with about 10% of HCH isomers. And this is just an image on the right-hand side of the landfill that was constructed on site for this kind of waste. So at least the first part, let's say, of the problem was or is being solved. Now, diving into our activity, this is the flow that we defined for our experiments and testing. So starting from sampling, isolating strains and microbiota only grown on HCH, analysis, detailed chemical analysis in order to understand byproducts and degradation products, then applying metagenomics and statistical analysis with bioinformatics, and then move into lab scale degradation tests and the pilot scale, and always metagenomics following these activities in order to understand what's going on, who's doing the job, and who's capable of degrading these contaminants and the isomers. And of course, toxicological assessment of the matrices. So as I said, this has a timely approach, and the first part obviously was the sample collection. There is quite a variety of contaminants also and soil types. The depth varied from 1 to 14 meters, mainly loamy sand and loamy clay soil type, and obviously co-presence of other contaminants such as DDT, benzene, and some hydrocarbons. So the idea was to select samples in order to understand also the interactions with other contaminants. So you can see it, I mean, from the first one, different HCH isomers, but similar soil type, identifying microbiota possibly associated with DDT, assess also the effect of a microbial community on chlordane or aldrin, and so on. This is unfortunately the normal, I mean the standard. It's rare to find only one contaminant in the soil. You always have a mix. So it is quite important to assess and understand the behavior and the response of microbiome to different contaminants, to different soil types, and so on, because this will help then define the best remedial approach. Some of the results, this is an ongoing work. So this was one of the selected samples with various isomers, but also DDT and aldrin and chlordane. And what we achieved was isolation of 3 fungal strains and 5 bacterial strains that were grown on only on HCH as a sole carbon source. And in parallel, we also established one mixed microbial community, microbiota, which is currently under test, still at lab scale, for possibility and capability of degrading HCH. The chemical analysis is also quite important in order to understand the degradation products and calibrate correctly the equipment. Mass spectrometry was used for this scope. Oops. So what are the next steps? Continue, as I said, the chemical analysis to accompany the degradation process and really understand the intermediates and the final degradation products. Metagenomics on the isolates in order to understand the degradation pathway and to perform taxonomical identification. So the idea really is to understand how the molecule degrades, how the ring is open, what are the degradation products, the end concentrations, and so on. Once this is done at lab scale, we will move towards pilot scales by using our RoboNova pilot testing facility. So just some images of the work, soil sampling, characterization, because this is also very important to understand from a physical and chemical point of view where the contaminants are, how it can be removed, DNA isolation for metagenomic analysis, and then microcosm or mesocosm test preparation. These are just some of the facilities that will be used. We call it soil testing facility. And on top of this, as mentioned, we will also use in a piloting phase our RoboNova unit that really allows to replicate a real scale biodegradation, on-site aerobic biodegradation. And basically it's a closed system. So imagine a bioreactor basically where you can control all the different parameters. You have a central unit where the soil can be loaded. You have fermenters that you can see at the back where we can isolate and reproduce our biomass. We can then inoculate back this biomass in the unit, and then we can start cycles, automated cycles. We can collect leachates. We can monitor the air emissions, we can treat the air. So basically we can engineer a system. This is the ultimate phase once we have understood all the behavior of the different parameters. We can add nutrients, we can add bulking agents, we can add water. So as said, imagine a bioreactor where everything can be controlled. And the goal of this is really to be able to scale up with great accuracy. And finally, I will touch again on the MIBIREM, although it was presented by Danny very clearly. But in particular, the part related to the objective of exploiting the microbiomes able to degrade certain contaminants. So the idea is to test single strains, but also microbiomes. We will mainly focus on bacteria in this work. It's almost a 5 years project, so a lot of data will be produced. Also prediction tools will be developed. And after a couple of years of testing with our partners from research institutions and universities, we will move towards the piloting phase. So we will have 3 pilots for the 3 different contaminants. We will have in situ, we will have on-site piloting. We will certainly use our RoboNova units. So again, the ultimate goal, and I think this is really important for this kind of projects, is to have some toolbox that can really be used and applied for research, for commercial purposes, for solving problems. So again, as I was mentioning at the beginning, we start from micro, we end up with macro. We study the soil, we study the water, we study the microorganisms, but then we start scaling up with lab testing, with pilot testing, with RoboNova, and in the end we have tools that can really be used by everybody that wants to solve the problem, because as presented before, and you've seen the numbers, the problem is quite widespread. And this kind of approach, nature-based solution, we really think for diffuse contamination in particular can be the solution. So this was my last slide. Thank you.
Good morning. I'm David Aguilar, the Technical Director of Biotechnology. We are a specialized company on biological formulations for the treatment of industrial water, municipal water, etc. We are very much focused on decontamination of the swine product, that is the slurry. And we always focus on decontamination. We have high versatility because we have very complex treatments, as you will see in some of the works that I'm going to present. And we also have other types of products that I'm going to mention. Well, we will start by focusing on pesticides. The intensive use of pesticides has led us to the contamination of waters, different bodies, water bodies. And this brings about problems for human, for human health, but all living beings. And also due to the accumulation phenomena. All these problems lead us to the search and development of treatment technology that allows us to minimize their impact on the environment. How are we going to do it? We can speak about chemical treatments, physicochemical treatments that are not biodegradable, that are persistent to the microorganisms' action. We can also speak about thermal treatments such as pyrolysis, vitrification, or incineration, burning, but it generates a high carbon footprint, and we are going to focus on biological treatments. If we can find contaminants or pesticides that are biodegradable, the treatment is going to be very efficient because we are going to achieve the adaptation of the microorganism to the contaminant. We can use biostimulation through the use of autochthonous microorganisms or the use of external microorganisms as a bio increase. And we are going to focus on that. Different new technologies have been developed at less cost. These are the bioprocesses, biological methods that are very efficient, not dangerous, not harmful, not toxic, affordable, that can be implemented immediately, that respect the environment, that is versatile because it can adapt to different situations, and that a method that does not leave any toxic byproducts. So the microorganisms consume pesticides to obtain the energy necessary for their development, eliminating these chemical compounds that are present in water and soil. The bacteria develop a genetic system against the toxic agents. And this is the focus of our treatment. And this is where we're going to focus our development in the adaptive capacity of bacteria to the environmental contaminants. So we're going to start speaking with our work with the LINDANE elimination unit. With the strains, we are going to look for an efficiency and method that could be efficient to remedy successfully LINDANE. And for this purpose, or to this purpose, we have 3 tests. The first one was aimed at looking for a stable population with different evolution through different generations. We used a tank, 2 tanks, sorry, Col 1 and Col 2, and at a stable temperature using contaminated carbon, Arlita, and we were using exactly the same mixture in the second tank, the cool 2. By the way, when I said Arlita, I mean expanded clay aggregate. And we were taking weekly samples in cool 1 and cool 2. And we wanted to generate a bacterial population that should be stable with low concentrations of HCH. We successfully adapted the bacteria to the lindane. It was because in these tanks there was air, so and this controlled temperature, and we could generate enzymes from the different bacteria and the different biological strains. We applied this on different receiving medium affected by lindane contamination. And the second test was we created more CUL cool, exactly the same as the first phase, but we created a third one in anoxic conditions with no oxygen, so as to see what of the formulations could create or be more successful. We tested 16 times in different containers at different conditions, different supports, and different contribution of bacteries that we were generating in our adaptive cool, the previous two ones. This is what we call the Blue Planet labs. And after the adaptation of the different bacteria and the culture stage, we started a new test phase in the lab with different volumes to control the degradative and adaptive activity. The third part of the test was to build bio batteries and adapt them to the soil to see the possibility of decontaminate the soils. And for this purpose, we extrapolated experiences obtained in different contaminant mediums where we saw that in aerobic conditions, we would be able, and with controlled temperatures, we would be able to demonstrate the efficiency that we saw in the tests, the previous tests in soils, and with a treatment upscaling these possibilities. And you can see the bio batteries with different blocks and different supports. So the expanded clay aggregate, also with some rice husks, and with at different temperature conditions. So we used 5 aerobic batteries, 7 anaerobic, so without oxygen batteries, and 2 mixed batteries. We also constructed or built 2 transparent shags to maintain the temperature conditions, to keep the dosage of the biological formula in every one of the bio batteries. We created more CULs, more cools. And as the moment we detected there was intestinal bacteria present, for example, in cows, we applied cow slurry in some of the tests, and the sludges from the Sardas landfill, we wanted to think that the bacteria that would be present there would be more than adapted to lindane. And therefore, we were playing a little bit with this mixing or combining formulations with endogenous and commercial and mixed purposes. And now I'm going to present the results. In the first test, along the different weekly contributions, we saw the efficiency when it came to reducing HCH. We didn't only adapt and be stable in a contaminated medium, but we performed a very important degradative line here with the bleeding and the 400 liters of the coal 2 that we were withdrawing, we analyzed that, and the results obtained were over 90% success, and this led us to the phase 2. Let's see the results of that phase. As you see here, we were having different cultures, an aerobic cool, cool 2, which was the one that we generated with the biological contribution of the stabilized bacteria, and the cool 3. In all tests, efficiency is more than demonstrable or provable in aerobic conditions as well as in anaerobic conditions and in mixed conditions. Lastly, the results of the bio batteries. Here you can see that they were very similar over time, and after different contributions, you always find very high results or very high numbers. The red ones are the anaerobic ones. You see that the results are not so good as in the aerobic ones, but in any case, we're talking about something that is really significant. Lastly, our mixed biobatteries, we can see similar results. By way of conclusion, I would say that all the results were rather promising in the end, as you saw from the first phases of our work. The reductions were over 90%, knowing that we were very efficient in the organochlorine part. And if we see the conclusions of the third phase, we can prove that we can work as a an enhancer of other endogenous strains. In the anaerobic part, we can see that the sludges by themselves and the slurry, we had worse results here, but they were the best ones when we put them together with our external biology, by external bacteria coming from our formulations. So the secret lies on the adaptive method, taking our bacteria, contribute different contaminants and pesticides or HCH or hydrocarbons, and make a genetic adaptation so that our bacteria generate a stable population, and in a hostile environment, they will favor gradually their adaptation mechanisms. So we will end up with a very efficient product. And we can apply that to any process in the soil. We can have an a la carte kind of biology, an a la carte bacteria, let's say, with more or less complexities depending on the demands. Thanks a lot for your attention.
Good morning, everyone. Today I'm going to talk about lab studies that lead us to make decisions in order to apply this into a pilot scale. So we all know by now how these HCH or other chlorinated pollutants can arrive into the groundwater and accumulate as a DNAPO. But fortunately, there's different strategies, chemical and physical strategies that we can use to solve this problem. But there's also the biological alternative, that is bioremediation, is why we are all here now. And I once heard someone say that bioremediation is the biology to the rescue. And I really like that, but besides all the advantages that we already hear now, there's one that I wanna point out, that is the high specificity that these microorganisms has against the pollutants, and how they can degrade the pollutants under the established, the legal limits established. So this is, for me, is one of the best advantages, more than the economical advantages and other that we already know. There's also plants, fungi, or other organisms that can bioremediate, but today I'm going to focus on bacteria, we know that there's aerobic and anaerobic bacteria, and they can use these pollutants as a carbon source or as an energy source, or just co-metabolically without gaining any benefit from it. So we from the University of Barcelona have been working together with Engrisa company in the last years. In the Sabiñánigo case, that the ones that were here yesterday know a lot about this case already, so I will skip that part. And our purpose was to study the feasibility of the bioremediation, the polluted groundwater, and to see which will be the best alternative aerobic conditions and aerobic conditions to treat this groundwater. We focus mainly in the alluvial groundwater of Sardas. So we chose here from the intermediate zone of the plume, we chose 3 main wells that we have been working. The average temperature of these wells was around 13 degrees. It's important because we're gonna be working at this temperature. And the redox potential of one of these wells was already a negative potential. That was interesting for us to try that anaerobic bioremediation, for example. And we have here the concentration of the pollutants. We can see that they are different in the 3 wells that we selected, less concentrated and a more polluted ones. So the first microcosm setup that we did has a purpose to determine if there was biodegradation occurring in natural conditions, or we needed to biostimulate, and if you need biostimulation, which will be the best strategy. So from this groundwater, we selected 3 wells, and we work with 13 degrees in the 3 microcosms, but in one of the wells, we also wanted to test at 25 degrees. We tried the natural attenuation, that is mainly the groundwater from the well, the aerobic biostimulation that had oxygen saturation, anaerobic biostimulation that contained lactate as an electron donor, and a treatment train that consisted in an aerobic treatment for 85 days, and at the end of this period, we did a saturation with oxygen. After this, we wanted to know the organisms that were present, so the DNA was extracted, and we did an 16S sequencing. Ooh, okay. So the first results of this, I'm gonna show just for one well here. We can notice that the aerobic treatment showed complete degradation of the less chlorinated compounds in a very short period of time. In 55 days, we already have all these monochlorobenzenes and benzenes degraded. In the treatment train, we just see degradation once the oxygen was added. In the anaerobic phase, we don't see. And for the natural attenuation, that was interesting to check here, we don't see changes after 85 days. So it was very similar to the control. I don't know if I'm blocking here. For this natural attenuation, we basically don't see change after this period. So for other compounds like the dichlorobenzene isomers, we have the same tendency here. The aerobic treatment worked better. Complete degradation of some isomers already at 30 days. And the natural attenuation also behaved very similar to the control. The control here is an abiotic control without organisms. Well, they were killed, basically. And for the 3 chlorobenzenes, we have the same behavior as for the monochloro and dichloro as the less chlorinated. But once we check the HCH family, we can see here for the LINDANE, for example, in blue, that already at 15 days, we have a complete degradation of LINDANE in anaerobic biostimulation. So for the most chlorinated compounds, we see a faster degradation in the anaerobic treatment. But the good news here is that also aerobically, we degrade this LINDANE or other isomers in a longer period of time. The LINDANE, for example, at 85 days, aerobically, we also degrade it. As I said, I just showed the results for one of the wells. The other two wells behave very similar, and the two temperatures also show very similar results, probably because the microorganisms were already adapted to these aquifer conditions. So I'm gonna focus on just one well. Once we got these results, we wanted to see if we could improve the degradation rate, and for that we tried to add nutrients. So we chose two wells. We incubated them at 13 degrees. We skipped the natural attenuation as we saw that was not working in a short period of time. And for the aerobic biostimulation, anaerobic, and the treatment train, we try adding phosphorus as a nutrient, nitrogen, and the combination of phosphorus and nitrogen. We here also reduced the time. We were working with 15 days instead of 85, so we wanted to see if in this period we can see a degradation and an improvement. And after this, we also analyzed the microorganisms present. So here for the less chlorinated, very nice results. We have for the monochlorobenzene and dichlorobenzene, the aerobic treatment. Once we have phosphorus added, or the combination phosphorus plus nitrogen, at 3 days, we have a complete degradation of these compounds. But we don't see this improvement when is only nitrogen added. At 15 days, we don't see the degradation. And in an aerobic treatment, we also don't see that. For the most chlorinated, let's see for the LINDANE. As we saw before, the anaerobic treatment worked better, was faster, and here is the same. At 3 days, we already see 100% degradation of LINDANE when phosphorus is added and in the anaerobic treatment. But good news for us, when we had nutrients in the aerobic treatment, we also have a degradation in 15 days in the aerobic conditions with phosphorus and nitrogen added. The same was observed for the summatory of this HCH that worked better and faster anaerobically, but also aerobically. Here in blue, we can see these results. So now I will combine these first results of these microcosms with nutrients and without. For the monochlorobenzene, for example, here is without nutrients, 50% degradation in aerobic conditions, but once we have nutrients, in 3 days we already see a complete degradation of the less chlorinated. One interesting compound is the 1353 chlorobenzene that was not degraded at 15 days in either the aerobic or anaerobic treatment, but once we added nutrients, the degradation already starts in all of them. Here I'm just showing the nutrients, phosphorus and phosphorus plus nitrogen, because nitrogen alone we saw that did not degrade, so just to clarify. So here for the LINDANE, it's interesting to notice that without nutrients here, we just have degradation in aerobic conditions of 100% in 15 days. But once we added the nutrients in the aerobic conditions, we have a complete degradation also in the aerobic treatment. For the epsilon HCH, that was also very hard and difficult for us to see the complete degradation. We see already a 10% degradation at 15 days when nutrients is added in the aerobic biostimulation. So here is an overview of the contaminants that have been showing by now. Where here's the aerobic biostimulation and the percentage of degradation, and here is the aerobic biostimulation. We can see just in a quick look without reading anything that there's a lot more bars here. So there's a lot of more degradation of these different compounds in the aerobic biostimulation compared to the anaerobic biostimulation. And the interesting thing here is that with anaerobic biostimulation adding nutrients, we improve the degradation rate of LINDANE and HCH family, but In the aerobic, we also improve these results when we add phosphorus as nutrient. So we see the degradation of lindane and the summatory of the HCH in a very short period of time, because it's 15 days. It's not so fast, but for us, it's still fast. So the microorganisms present, we just wanted to show here that at initial stage, we already see some phyla like Chloroflexi, Firmicutes, Protobacteria in the populations that could include microorganisms that are described as degraders, also for aerobic and anaerobic strategies. So the interesting point here is that at the initial stage, we already have microorganisms adapted. After the aerobic experiments, the aerobic biostimulation, we see also a great bacterial diversity, including degraders of HCH and lindane that are described in the literature, as can be Pseudomonas, Acidovorax, Sphingomonas. We are not saying that these are going to be the responsible. We just say that they are present, and they could be more than one. We don't know that. And for the anaerobic biostimulation treatments, we also have here a percentage of Delococcoides, Delogenomonas, Delobacter that have also been described as HCH degraders or chlorobenzene degraders. And they are in a very low percentage. This is a relative percentage of the overall bacteria in this groundwater. And we have to remember that these anaerobic bacteria are very slow-growing microorganisms, so that's why probably they are very low percentage. But the good news is that we have potential degrading. So to conclude this presentation, we can say that the bioremediation proved to be a suitable strategy to treat the alluvial groundwater of Sardas, that natural attenuation conditions did not show significant results, therefore biostimulation is really needed, that the aerobic biostimulation degraded the less chlorinated faster, but also a longer period of times we can degrade the most chlorinated as lindane and other HCHs, and that phosphorus addition improved the results of the degradation of most of the pollutants after 3 days, that the addition of only nitrogen did not improve the degradation, the presence of chlorinated degraded microorganisms also in aerobic and anaerobic conditions were identified, so we have microorganisms there that could work for us. And the final conclusion, or for us the main one, is that we can propose that the aerobic biostimulation with phosphorus addition is a good strategy to apply in situ in this special case to degrade lindane and other chlorinated pollutants. That's all. Thank you for your attention.
15 minutes.
Our next speaker is Dr. Kevin Kuntze. He's an innovation expert at ISO-Detect in Germany.
Greetings from Leipzig, Germany, and I'm sorry that I'm not in person there, but maybe next time. But still, many thanks to the organizers that still have the opportunity to present another tool to assess the biodegradation. It will be a tool that will be assessed in situ biodegradation. So many thanks also to my colleagues, Danny and Cosimo, and also Jesica, who really nicely introduced already into topics of natural-based solutions. But now, when we assess the field sites of HCH contaminated sites, we have already confronted with many different questions. So who was this? Who was this? Who's responsible for this HCH contamination? Maybe there are more than one responsibilities for this, so more sources of HCHs. Is degradation already taking place? And can we detect this already in situ? Or can we also stimulate this degradation? And can we monitor the stimulation of degradation? So as I already said, the previous talks already nicely present some toolbox like qPCR analysis, metagenomic analysis, and for sure also concentration analysis. And another tool to really assess in situ the biodegradation and also source allocation is the compound-specific stable isotope analysis, what I would like to focus now. So in general, as an introduction, what are isotopes? Isotopes are atoms of an element with the same number of protons, but different number of neutrons. So you have here a periodic chemical table. As you can see, the number of protons, that's why you have different elements. But each element could have also stable isotopes, for example, shown here for the carbon, and also for sure radioactive ones. But we are now talking only about the stable isotopes. So they are not radioactive. So for example, the carbon has two stable isotopes. It's the 12C, so we have 12 neutrons and 12 protons, and the 13C. And the 13C has just one neutron more than the 12C. That's why it has the different masses. And the 13C and the 12C carbons are natural abundance within the organic compounds worldwide. So, and the natural abundance of this is about 1% of the heavy carbon of the 13C to about 99% of the light carbon, the 12C. But this more or less 1% to 90%, this more or less is really important because the small differences of 0.96 to 1.16 is really important to measure later on for source identification and also for the proof of in situ degradation. So what we now will measure, we will measure the carbon isotope ratio. So the ratio of the 13C to the 12C of the organic compounds. This you can do also for other elements that have two stable isotopes. For example, also the hydrogen or the chlorine is also possible for ACH, or nitrogen, oxygen, or sulfur when you have other compounds. Measured, this ratio, for example, the carbon isotope ratio is measured via an isotope ratio mass spectrometry. And at the end, I'm talking about delta values. And this delta values is the ratio of 13C to 12C given in per mil. And it's just a value, as it's the ratio of the sample measured according in relation to an international standard just for comparison that it's independent from which lab it was measured. We have an international comparison to a standard. And this is the value at the end, what I'm presenting. So each organic compounds can consist of many different elements. For example, here the MTBE, yeah, you can have carbon, you can have hydrogen, you can have oxygen. Also the situation for ACHs. And when you measure now the ratios of the stable isotopes of each element out of these compounds, you can have several. You can have a delta 13C value, you can have a delta chlorine value, a hydrogen value, oxygen, and so on. And for those organic compounds that have been produced, it's important they have really a distinctive isotope ratio due to their production line and also the time they were produced. That's why really we have with a compound-specific stable isotope analysis a tool to do fingerprinting, to really do source identification. And sometimes you can also have a time estimation when there was spilled, but really you can have really a proof of source identification. A second really tool, or the question, why do I need to analyze them, is beside the fingerprint, you can have as a proof of biodegradation, because pollutant molecules that consist of a heavy isotope are degraded more slowly than those that consisting only light isotopes. This is shown you by the example of ACH. So you have one molecule, so with HCH that have a heavy isotope, and the other one have only light isotopes. And when they are degraded by microorganisms, the degradation of those with a light isotope is slightly faster than this degradation of where they have a heavy isotope in the molecule, because to have a bond cleavage here with a 13 carbon needs more energy. That's why it takes more time to degrade those. So within a pool where you measured several compounds, so just imagine you have now several 12CHAHs and some 13CHAHs. And this pool we are measuring at the beginning and then during degradation. We would see, because first only the light isotopes, the molecules with the light isotopes will be degraded, we would see an increase in the measuring pool of the 13C in comparison to the 12C. And that's why your isotope ratio is shifted towards more positive isotope values, yeah, within the contaminant degradation. So this is really can be applied for really many different organic compounds, contaminants like the BTEX compounds, naphthalene, the chlorinated ones, but also for ACHs. So in blue, you have here the source signature. This is due to the production line. So each can have different primary isotope signatures because of their fingerprint, because of their production. But when those HCHs have been degraded, the isotope ratio will be shifted towards more positive isotope values. And this I want to show you now for a certain field site where we applied this method. It was the HA8 contaminated site, as you can see here, with a western fringe, you have an eastern fringe, but you have an upstream flow and then also in central flow. So the groundwater flow direction was here from the well A towards the direction of well C, D, and then more in the direction to the well E and F. The ACH contamination was about 100 microgram per liter, and we took several samples over 3 years, and we would like to focus on 2 main questions. Can source zones, that has been predicted here because due to the concentration, but can they be really differentiated between each other? So can source zones of SCHs be confirmed by compound-specific stable isotope analysis? And does in situ degradation of SCHs already take place, and to which extent? So those are the results for the delta SCH. So this is the field map, as you can see, that we know at which well we are. Here we, as I said, we took samples over 3 years from different wells. In the gray bars, you do see the concentration of the ACH, of the delta ACH on the left side, and the black dots are the isotope ratios of the delta ACH. So starting at well A, because here this is the groundwater flow from the upstream well towards the groundwater well A, you do see we have an increase of the delta ACH concentration. And at the same time, we have a relatively negative isotope value of about -28 per mil. And that's why we have already an indication that yes, we have here at groundwater well A first source of contamination. And this was confirmed by a former production by the vicinity of a former production facility at well A. Going then from well A towards well C, from B to C, though in this direction, because this is the groundwater flow, and on this groundwater flow, we would expect maybe if degradation takes place. You do see that independent from the concentration, okay, we have in degrees, but you have also some fluctuating concentration. You do see a significant increase of the isotope ratio. So we have a shift of around -28 per mil towards more positive isotope values of about -24.5 per mil. And this is really a proof that degradation in situ degradation takes place, because this shift of the isotope ratios can only be due to degradation processes. Any other physical processes like sorption, like dilution or evaporation cannot really affect the isotope ratio. So if we do see a shift in the isotope ratio, we have a proof of degradation independent on the concentration. So we can really say, okay, we have a decrease in the concentration here, but really we can say, yes, but this is also due to degradation and not only to physical processes. Furthermore, then when we reach the well D and well E, as you can see, we have really high concentration again of delta SCH. And we have a drop down of the isotope values to really negative isotope values of about -30 per mil. And the shift towards more negative isotope values is not possible to any degradation of physical processes. That's why we have here a proof that we have a second source of contamination. And this was confirmed by a former HCH dump site at well DE. Furthermore, if you go then from in the groundwater flow direction from well DE to F, you do see again an increase in the isotope ratio. That's why we have here also a proof downstream from the second source of contamination, we have also in situ degradation of delta ACH. This was also the situation at the western fringe and the eastern fringe. In comparison to the source isotope signature, we do see a shift in the isotope ratios towards more positive isotope values, and therefore we have also here a proof of an in situ degradation of the delta ACH. For the gamma ACH, so for lindane, we have not, the concentration was relatively low at all wells. But we will now focus only on well 3 that was not established in the first year, but in the second year. And as you can see, so it was here, we have really high concentration of the gamma ACH. But in comparison over the year, so in comparison to the next year, we see that we have an increase, we have a shift of the former isotope value of about -27 per mil towards more positive isotope values of about -25 per mil. And therefore, we have also a proof for the gamma ACH that it's been degraded. And also we have here proof of the high concentration and relative negative isotope values that we have here a third source of contamination. So with applying this compound-specific stable isotopes, we significantly improve the conceptual site model. We were now able to show that, yes, we have 3 distinctive sources at a site, and we really have evidence of biodegradation taking place downstream from well A, as well also from the second source of contamination around from well DE to F, and also in the fringe and the western part. Furthermore, and this is really the only method where it's possible, we can quantify in situ the ACH degradation. For this, you need a Rayleigh equation. As you can see, it's relatively easy. Those are the values from the field site. So this is, for example, you take as the source isotope signature. And this is the delta value of a downstream well for which you did see a shift towards more positive isotope values. A second parameter you would need is the isotope enrichment factor epsilon. And this epsilon reflects or presents how fast or how to which extent such an isotope fractionation is taking place due to degradation. And this depends on the microorganism catalyzing those degradation and also under which conditions. So in this, of course, there are many literature data given. You can have also, we have a database also where you can use those isotope enrichment factors, but you can also determine site-specific isotope enrichment factors by lab experiments, what Jesica also nicely shows. So some lab experiments, you do degradation, and beside the concentration analysis, we will measure also the isotope ratios, and therefore we have the enrichment factor. And by taking this without any concentration, we can determine the in situ degradation rate constants, first order degradation rate constants, distance related and time related. So, and as you can see as a conclusion, with the compound specific stable isotope analysis, we confirmed different source zones at the investigated field site. We provided evidence of SCH degradation in the investigated aquifer, and we allowed also possible to quantify the in situ ACH degradation for the expected flow path. And with this, I want to thank you for your attention. And yeah, if you're interested in this, you can just later on, there will be discussion, or you can directly contact me. Thank you very much.
Our next speaker is Dr. Juan Escobar-Arnanz. He's a chemist with AECOM in Madrid.
Good morning. In some other presentations that will be presented today, we will speak more in depth about a bioreactor, but just to give you some context, I'm going to describe what is such a system. This is a biotechnological system able to provide controlled and isolated conditions that will maximize and enhance the growth of a culture of microorganisms that carry out the degradation of the contaminants of interest in the aquifer. Of course, this is what interests us here. A bioreactor, therefore, has to be a system providing a continuous source of microorganisms to the environment, or to the medium rather, but it has to be efficient, robust, and should provide control conditions to ensure the efficiency of this system until we get our goals. Previous to the application of a bioreactor on the field, normally it is necessary to conduct a previous kind of study or work to ensure the good working conditions of such a system that rely on a lot of work, laboratory work. However, this process and this transition is not a simple one because there are many factors involved from the physical, chemical, and microbiological points of view. And therefore, for this transition, in many cases, it is necessary to carry out a escalating process where we or scaling up process where we play with the different parameters that affect bioremediation to ensure the correct operation of such a system. A good starting point in this process is the obtention of a good bacterial starter or inoculum that is a starting point, and the microbial community is going to be adapted to it to increase the degradation capacity of the contaminants of interest, which will determine the biomass that will be feeding the bioreactor as the bioremediation mechanism. With a good analogy with cooking, for a microbiologist to have a good inoculum, it would be to have a good dough for the bread making. And the better the raw materials at the starting point, the more successful the possibilities at the end. And therefore, for a good work, the obtention of a good inoculum for a bioreactor is key, is essential. In this work, we wanted to characterize different samples that have been affected by different episodes of contamination by HCH in order to assess their possible use as inoculums or inoculi for inoculants for the development of an in-situ bioreactor. We used 13 indigenous samples from the Bailin landfill so as to create an internal kind of library, and we wanted to select the different nature of water, soil, and contaminants. Every one of the soils were distributed in different areas of the Bailin landfill, and they were possessing different nature in terms of water, soil, sediments, and sludges as well among the samples chosen. Every one of these was characterized from the microbiological point of view, but we also conducted determinations to get to know the contaminant burden, the nutrients, and the physicochemical characteristics. As for the microbiological characterization, we selected 4 attributes determined through different lab tests that were important for the characterization of the samples and for the next steps. The first one being the determination of the cultivable and viable biomass in generic medium, which would determine the plate count. And the second one would lead us to determine the viable and cultivable biomass in minimal medium, having HCH as the carbon source. And we want to determine what are the microorganisms that are specialists in this contaminant. The third test is focused on determining parameters related with the microbial activity of microorganisms and the diversity of the strains. And the fourth one is an experiment to test the effect of different concentrations of contaminants on the metabolic activity of the bacteria in the samples. Every one of these determinations was carried out according to the flow of work that you can see on the slide, taking into account that it was necessary to leave 3 weeks from the sample collection to the sample preparation and the monitoring of the samples. Here you can see some of the photos of the different phases of the process that I'm describing. Now I'm going to comment on some results very briefly, especially the microbiological characterization that we carried out. As for the determination of the viable and cultivable biomass in the generic and in the minimal medium, we counted plates after the sowing and an incubation period, depending which varied depending on the cultures used. The results of this stage allowed us to observe concentrations of microorganisms between 10 to 3 and 10 to 5 in all the samples, being these latest values very interesting values to carry out future bioremediation experiments in the Bailin landfill. It is interesting for us to see that concentrations in both the generic medium and the minimal medium were very similar in both cases, which indicates a high degree of specialization of these micronutrients or bacteria in this medium. As for attributes 3 and 4, that allowed us to determine the metabolic activities and the microbial diversity. And the fourth one, the tests of the metabolic activity before different contaminants. We carried out the tests in plates with 96 holes or wells, small wells in the plate. And the colorant was being reduced to create a purple kind of compound, which corresponded to the metabolic activity of microbes. So the more metabolic activity by microbes, the deeper the purple color. And here you can see how we explain the life cycle of microorganisms when they are facing a new medium. Microorganism will have the lag phase when it adapts to the new conditions of the medium. Once it is adapted, it starts getting nutrients and substrates present in the medium to experience some growth and proliferation that is made exponentially up to a table phase or a stationary phase, and then the death, when the number of microorganisms are reduced, either because there are not enough micronutrients or the presence of metabolites that can be harmful for the microorganisms. The goal of these experiments on the plate was to monitor the color development a long time, so that if there was enough rate of metabolic activity, we could see these growth curves that would be mimetic with the behavior that we expected. Here we used commercial plates. The first one is the ecoplate plate or microplate that allowed us to see the microbial activity, and the second type of plates when these microbes were faced to different contaminant concentrations. Here you can see some of the microplates used for the test for different samples that we included in the study. As for the different metabolic activity experiments, we obtained these curves that you can see on the slide, where we can see that for most of the samples, we obtained growth curves with a behavior that was mimetic with the theoretical expected behavior, with short growth phases, long exponential phases, and the stabilization. From these experimental curves, we were able to get different parameters related both with metabolic activity and the diversity of microbial communities, as the duration of the lag phase or the growth phase, or the different indexes according to the different substrates. We can get different indexes such as the Shannon and the Simpson indexes that, according to the sample, were having values over 3 and over 0.9 respectively, which show the different values of microbial activity. As for the last test, the microplates were configured to allow us to test up to 7 growing concentrations of contaminants present in the aquifer. The goal of this study was to get experimental curves that would allow us to differentiate and define what concentration of contaminants would be ideal to maximize the activity of microorganisms in the samples. As for the experimental curves obtained, in this case, we can see higher differences in the different samples than what we saw with the echo plates, and with lesser metabolic activity, which is logical because HCH is a much more persistent substrate, more biodegradable than the ones that are used substrate in the echo plates. By way of conclusion, all these determinations and criteria that we have seen were assessed globally in order to determine whether there were interesting candidates for the following steps and to choose them according to the different characteristics and choose which ones would be more interesting for which steps of the study. We were able to choose 6 samples out of the 13 that were included in this work, according to the different biomass values, the diversity of their microbial communities, and the tolerance presented to different concentrations, to the concentrations of the interesting contaminants, or the contaminants of interest for our study. Thank you very much for your study, for your attention. Sorry.
Okay, our next speaker is Elena Granados, and she is an environmental scientist who studied here at the University of Zaragoza and is now working with EMGRISA in Madrid on soil and groundwater pollution.
Hello. Firstly, I would like to say that this work that I'm going to present has been done jointly with EMGRISA, with Sarga and the Government of Aragon staff. And we are going to speak about the characterization of naturally present microbial populations at Sarda's landfill and the Inquinosa factory in Sabiñánigo. As many of you know, microorganisms present in an ecosystem contribute many ecosystemic services. They decompose organic matter, they put nutrients at the disposal of other living beings, and they are able to take contaminants present in the medium and produce bioremediation or a high degree of diversity in a biocommunity will provide greater resilience and adaptation to disturbances, and they also are in charge of primary production. In this work, we wanted to assess the status of the native bacterial communities in order to, on the one hand, know and get to know what is the biodiversity and explore bioremediation techniques. The study areas were 2. The old Inquinosa factory, as you know, is the source for the HCH contamination in the area of Sabiñánigo and the Sardas landfill. In total, we took 37 samples. In Inquinosa, there were 8. 4 were soil samples and 4 groundwater samples. In Sardas, we took 29 samples, 8 from soil, 7 from sludge of old pond, and also 6 from groundwater samples, 4 water samples of old pond, and 4 vegetation samples. What did we do with these samples? On the one hand, we made a physicochemical characterization determining the pH, the conductivity, the content in phosphorus, nitrogen, organic matter, and also chlorinated organic compounds, COCs. And then we carried out a microbiological characterization. We determined the total cultivable biomass and the cultivable specialized biomass, specialized in decomposing chlorinated compounds. Also, we wanted to assess the community parameters, ecological and metabolical, and we carried out a toxicity test. In terms of the determination of the total cultivable biomass, we created a colony count in generic culture medium, and also we used another medium with DNAPL coming from sardas as the only carbon source. Then we used biolog ecoplate plates to determine parameters that were characteristic of these communities, the latency time, the metabolic time, as well as ecological indices, the Shannon, Simpson, and Shannon, again, diversity index. We also use the MT2 microplates prepared with different DNAPL coming from sardas concentrations. The most important concentration contained 35 parts per million of HCH. What were the results? Firstly, the counts of microorganisms were high, around 80%, in around 80% of the samples. That is, the samples are rich in bacteria. The lower results were presented by water samples, not all of them, but all the low results corresponded to water samples, and the lower ones with 10 to 8 values were the highest results, 10 to 8, were vegetation samples. And in 38% of samples, we detected more growth in restrictive medium plates than in general mediums, although the colonies were more diffused. As for the values obtained with the biolog ecoplate, in 3 samples, it wasn't possible to conduct a study because the metabolic activity was really low. As for the latency time, in the rest were between 12 and 46 hours, although in 76% of the samples, the metabolic activity was carried out in less than 24 hours. As for the Shannon and Simpson diversity indexes, you can see the slide. As for the toxicity test, we recorded less metabolic activity in the MT2 respect to the ecoplate plates. In the case of Enchynosa, 6 out of 8 samples were presenting significant metabolic activity. In the case of sardas, only 4 out of the 36 that we studied, we chose 2 as the ones presenting not promising results. The M19 Enchynosa sample corresponding to the S21 borehole, and the M333 sample from sardas, which corresponded to root sample. What can we see in these results? We can compare how these 2 samples make use of the different DNAPL dilutions as the carbon source. In the case of the M19, it's registering more or presenting more metabolic activity. And here we obtained it with the 150 maximum activity. In the M33 sample, we see highest toxicity resistance with the highest metabolic activity, which decreases as the contaminant concentration is decreasing. Conclusions. On the one hand, the microorganisms that we have in the 2 sites, despite the contamination existing, are perfectly well adapted to the environmental conditions. Despite the fact that the plates in a restrictive medium are growing well, it indicates that in a restrictive culture medium, it indicates that they have no competition, and therefore high growth. And therefore there is a good metabolic activity. Therefore we recommend biostimulation techniques. As for the M19 and M33 inoculums or samples, we consider that they could be good candidates to conduct future tests in bioreactors and to use them as bioinoculums in bioaugmentation techniques. Thank you very much.
Okay, and for our final speaker before the break, we have number 2 from Dr. Escobar-Arnanz.
Hola de nuevo. Hello again. As we saw over yesterday's session, normally in sites that are contaminated by HCH and DNAPL, resulting from the production of the pesticide, is that during the early stages of remediation, treatments with physical and chemical strategies are applied to achieve a reduction of the concentration of contaminants present in the site. Once we significantly reduce the parameters, some cases it is possible to think of the transition towards bioremediation treatments. These bioremediation treatments, in order for them to be efficient and viable in contaminated sites, they require certain time. In many cases, they are not easy to guarantee, their success. In many cases, it is commendable to characterize in detail in order to collect different lines of evidence from different viewpoints, for instance, from the physical, chemical, and microbiological viewpoint. Among other aspects, these types of characterizations allow us to collect evidence about the microbial communities and degradation pathways existing on the site, so that biodegradation conceptual site model can be built, allowing for an assessment of bioremediation alternatives that can be successful in a specific aquifer or contaminated site. For these characterizations to be carried out, there are different alternatives. For instance, one option is to carry out laboratory studies based on cultivation methods that are very interesting and very useful in order to carry out studies in depth, allowing to determine and adjust variables and parameters related to biodegradation of contaminants, of pollutants. However, these type of techniques only allow to grow 1% of the microorganisms in the laboratory. As an alternative, we can also carry out in situ characterizations using molecular biological tools and isotopic analysis techniques that allow to analyze micromolecules and organisms in the molecule in order to increase the representation of these molecules and reduce uncertainties related to the other techniques. These tools are a set of tools or techniques, both analytics and biochemical, that allow us to analyze biomolecules of the natural or environmental or site sample to get an idea of the activity of the microorganisms in a contaminated site. From then on, we can answer questions such as what microorganisms are present in the site, what is the concentration of the degradant microorganisms, and if biodegradation processes are happening in the site. With this sequential application, therefore, it is possible to collect a more clear idea of the microbiological microorganisms present. And for that, we can use different sampling methods adapted to the goals, such as specific samples for specific place, and also passive samplings for a specific time frame. And with the use of these tools, it is possible to get different sorts of data and for different goals. As we have heard before, one of the main applications is to understand what are the microorganisms and biodegradation processes in eventually to connect it to the biodegradation and the microbiome existing in the place. But these techniques are also used to assess temporal and spatial changes in these communities that could be linked to specific action or work in the site. It is also interesting to use them to evaluate different sorts of amendments and try how these affect the activity of the microbial activity in order to determine what the treatment could be in the aquifer. In the Bailin aquifer, these techniques have been applied from 2017 up till now, and in different places of the site distributed as you can see here in this image. Then we're going to see 3 applications of these techniques over this time. The first is about the application of next generation sequencing. This allows for a non-directed analysis, non-targeted analysis, to have an overall profile of the microbial community. This allows us to do a classification of taxonomies, also gets an idea of the biodiversity and identification of microorganisms or predominant degradation processes. Sometimes, depending on the concentrations of microorganisms, sometimes determinations can be at the level of species. This study took samples from 22 boreholes with different hydrogeochemical characteristics. We could obtain compositions of the microbial communities, and we saw significant differences in 3 of the boreholes of the 3 different zones. We also identified material from different genuses that might be linked to the presence of contaminants of COX, such as the Pseudomonas, that could be related to HCH degradation processes and others related to benzenes. The results of the applications of this technique also allowed us to obtain different indexes or rates of diversity, such as the Shannon rate, as you can see here, a Shannon index. And it is interesting how samples with higher diversity were not necessarily the ones showing higher concentrations of total bacteria. The fact of having more diverse microbial communities is interesting because sometimes this is linked to a higher versatility or availability of degradation organisms connected to COX on a site. In this study, we could also analyze main components analysis that revealed significant differences in the microbial communities, such as those coming from higher concentration boreholes and samples from boreholes with less concentrations of COCs. Also interesting is to see how boreholes belonging to the same area of the site were also showing results, very similar results that can be seen here. In the second case study, it was about the application of qPCR for the analysis of several biomarkers of DNA related to certain biodegradation processes. Through this technique, we can quantify these DNA markers, thus determining presence or absence or a functional gene related to particular degradation process, and eventually related to potential for degradation present on the site. With this technique, we can analyze several DNA markers, such as the one shown here related to certain degradation processes of different COCs, both anaerobic and aerobic pathways. We highlighted those related to the compounds present in higher concentrations in groundwater in baleen, such as benzenes, chlorbenzenes, chlorophenols, and HCH. From these concentrations of these functional genes analyzed in this study, we could obtain biodegradation potentials for the different compounds and under different conditions. It is interesting to highlight that we saw biodegradation potentials high and medium under anaerobic conditions in several boreholes in the site. And the last study case studied quantification of RNA biodegradation using this technique, RT-qPCR targeted quantification. RNA is a result of the transcription of DNA as a prior phase to the formation of protein. Through the application of this strategy, we're able to identify these transcripts or molecules and saw their presence in a specific degradation pathway. We can thus get closer to the actual activity of a degradation process. However, it could be interesting to use proteomic studies too. Apart from biomarkers that we saw before in previous slides, in this study, we included some biomarkers that were related to aerobic degradation of lindane, particularly lin genes and lin A and lin B. In this case, we took samples of 6 boreholes all over the site with different characteristics, and we can see we identified concentrations of both DNA markers in solid bars in these charts, and also of the transcripts, corresponding transcripts that are shown also as lines of dots. We saw how at least in 4 of the 6 boreholes, we detected interesting concentrations of both the biomarker of DNA and the corresponding transcript of lin A gene of the degradation of HCH TCBs, but we didn't saw lin B gene. As a conclusion, even though we are still at an early stage in the baleen aquifer for the application of treatments of bioremediation, we have been applying sequentially these tools and isotopic analysis to assess the potential installation of bioremediation alternatives. Through these sort of tools, we could saw microorganisms and functional genes related to the degradation of the contaminants of interest in the aquifer. Also, diverse and adapted microbial communities have been detected at different locations on the site, and different lines of evidence have been collected to demonstrate the existence of biodegradation of the different contaminants at the site today. I thank you for your interest.
A question for both Elena and Jesica. Would you care to comment on the pros and cons of biostimulation versus bioaugmentation for HCH compounds?
I'll say something. I think that a best strategy could be the biostimulation, because is easier to apply at the field. Applying microorganisms, I think there's more things involved. You need to keep them alive and to grow them and then transport them to the field and then apply them correctly. And if it's anaerobic microorganisms, for example, there's more complication in the application to the groundwater. So I believe that the biosimulation has this advantage. You just work with the microorganisms that are already adapted to the conditions of the aquifer and also adjusted to the contaminants there that can also be toxic to them. So I think that's the difference and the advantage of the biosimulation.
Well, I think that biostimulation, at least in our area, I think biostimulation in our area can also be interesting, the problem of the contamination in our area. And I think biostimulation could be also easier to apply because we know that the microorganisms existing there, they act in a synergical way. And in the end, they are more efficient in the degradation exactly where they are already. And for us, as Jessica said, it's easier to provide nutrients to them, oxygen and conditions enabling them to carry out their activity there.
I have a question. I'm not expert on this field. I have worked on the role of microbial processes, and I am very interested on what these methods could give us. Now, my question is, how can you quantify the actual role of these microorganisms in degrading these compounds? So I heard you say well, you don't use the term remediation, but these microbes are doing the job of degrading these compounds. How can you quantify? Is that possible? Because then it will be possible to say how efficient we are in improving that, okay, going beyond that.
I don't know, maybe you can complement if I am short. I think we do this control of the contaminants on time, so they are being measured and you're gonna see the concentration. Of course, they are moving and there's all this problem. But there's one tool that somebody was presenting today that was the isotopic analysis that can help to see how fast you are moving on the degradation. That is a tool that helps in situ for you when the contaminants are adsorbed and then they desorb again, and then you have again high concentration in the groundwater. And in a field that you see the microorganisms are degrading, and then there's a desorption from somewhere, and then the concentrations increase. But with the isotopic analysis, I think is an extra tool that you can really use to measure this degradation, because the isotopic fractionation is going to increase.
So basically, I mean, what I think is that you have several tools to measure and control. I mean, certainly isotopic analysis. I mean, from a more qualitative point of view, you can have metagenomics to identify the species, and you can have a qPCR as well. You can have bacterial counts. So you are able to really identify and track both quality and quantity of microorganisms that are really responsible for doing the job, as you were asking.
I think I could add one other thing too. If maybe I'm taking the question in a slightly different way. Are you asking, is a specific culture or a specific strain of bacteria actually degrading the HCH compound? Well, one way to do that, something that we did years ago, was to use carbon-14 labeled LINDANE and then do a mass balance on where that radioactivity went, because there's a partitioning of about 60-40 between the compound going off as carbon dioxide and 40% being incorporated into the biomass of new cells that are growing on it. So you could use carbon-14 to prove that those organisms were actually degrading lindane and incorporating it into their biomass.
If there's another question, no, just adding, I mean, once you have the tools, one of the question is to understand if it's single strain or a consortium, a microbiota doing the job, and this is one of the objectives, by the way, of the MIBIREM project that was presented before by Danny, really to understand. So there are tools available as presented, as also Alan mentioned, but really the key is then understanding who is doing the job, if it's more single strains, if it's more consortia. And again, it depends a lot on the local conditions. I really think a targeted site-specific approach is needed because, as I also presented, there's often other contaminants present. I mean, it's rarely the case that you have pure, HCH alone in an aquifer or in a soil. So obviously the complexity is high.
Well, I'm not an expert on microbiology either, but what we have seen at sites contaminated with chlorinated solvents, particularly those sites where there is DNAPO, the halococoid have been great biodegraders, but under anaerobic conditions, which I guess is a challenge in the alluvial aquifer, for example, and Sardas, but also in fracture media, which is very difficult to achieve anaerobic conditions. So we need to inject a lot of electron donors, like lactate, in order to create these conditions. And something that I miss is that it's a map of the redox condition of these aquifers. We don't know where those anaerobic conditions are naturally located. And I'm wondering, for example, in your case, how much lactate you had to inject in order to create those conditions. Thanks.
We work in the lab scale. So we work in anaerobic chamber, in anaerobic conditions, with anaerobic bottles. And we added lactate, 3 millimolar concentration to produce this, to generate the electron donors that we needed. And we added, after sometimes, I didn't show that, but after 30 days, the lactate was consumed and we had to add it again. So yes, I think in the field scale, you need to really control the, measure what you have in there, and we are adding and then measuring the redox potential, yeah. And there's different, several wells that were studied, and some of them had already a negative redox potential, but some of them were very positive. And this is one of the ways that you can control or at least know the anaerobic conditions with using in the field, using the redox potential.
This is a question for Mr. Aguilar. You mentioned how you were using external microorganisms for bioaugmentation, but sometimes when introducing external microorganisms, adaptability to the medium, was it successful? Because sometimes local microorganisms can kill external microorganisms. That was my question. Yes, well, as said, we work with formulations on the liquid medium. So we have bacteria with higher potential of activations due to the latent conditions. We start with a mix of compounds or populations with different types of bacillo and nitrobacter and nitrosome. And this is added to a pool of biological nutrients coming from our laboratory in the United States. And we know the type of stabilizers that we are adding to the different strain. But when we introduce an adaptative method, we see exactly what is needed. Dosage is constantly being monitored, so we sort of deactivate potential killers. So in the end, we are able to get a very adaptive consortium together with endogenous bacteria. Our experience in wastewater is that there is always potential for survival. Of course, competency is there, but we can formulate different strains with different nutrients depending on our goals. If we want to reach other certain goals, of course, we need to study adaptations. We have experience in hydrocarbons, and we have many experiences with different contaminants. I'm not sure if I was able to answer you, but we have a good method working with the biological competition on the site.
I would add to that discussion that the converse is more likely to be true, that especially in soil, it's hard for the introduced bacteria to actually compete and survive. In groundwater, it's easier because the numbers are so much lower. You have a dilute system. But in soil, the introduced bacteria, more often than not, have trouble competing, and therefore they don't survive.
Okay, and this is for people working on this bioremediation or bio stuff. If we want to implement this in the field, this will be a huge challenge. Okay, be aware that those of us working on the dynamics of the flow and transport, okay, if we want to analyze how the chemical plus the biological processes, how they take place at a scale, my understanding is less than a meter or something like that. This will require models and quantification that we don't have yet. So you should be aware that going into the field will require further developments from other fields, hydrogeology, solid transport. Okay.
For sure. Maybe I can go ahead, Alan. That's okay. Yeah. You can. Okay. You can hear me. Yeah. Yes. Yeah. Okay. Sorry. Thanks for this comment. Also, for sure, I think at the end, it was already. I tried to answer your first question on which the proof if degradation is really taking place. I tried to step into, but now you give me the opportunity at the end. So. For sure, it's a combination of monitoring tools. For sure, you have to adapt also, you have to go for a hydrochemical analysis as well. But yeah, I would also like maybe go again on your previous question on the activity, but Jessica already nicely answered this question by isotope analysis. So this is the proof, and also I completely agree with also what Cosimo said, that at the end you need a combination. Yeah, you can do qPCR analysis in the field and combine this with isotope analysis. Then you have a proof that there's degradation going on, and you have hints who's doing the job. And then at the end, as also Jessica said, you can also go into the lab, and then you can do several lab studies in degradation, and you can do what Alan said about the 14th C. It's also possible to do this with 13th C. It's much not so critical and much easier, more sensitive. So this is also possible that you adding a labeled compound, labeled HCH, this we already did, to your microcosms, and then afterwards you can check also, you can also go for DNA, check for labeled DNA, and then you can identify who's the responsible community. But I don't want to go further this, so thank you very much.
And just one last comment, considering the consideration from the gentleman. You're right. I mean, there are many different factors that needs to be taken into account. So it's not only the microbiology, it's hydrogeology, is the knowledge of the contaminants, the concentration, how the contaminants is in the water or in the soil. So it's a very complex system, but as I was also explaining during my presentation, I mean, the idea is to go step by step and gradually upscaling. So from lab scale experimentation with micro and mesocosm towards prototyping and pilots. I mean, the idea is to consider all the different parameters and model them, also with some prediction tools that we are developing, and academia is also developing in order to reduce the uncertainties. But certainly all the parameters need to be considered. Otherwise, you will not have a full picture, and in the end, it will not work on a full scale. So this is very important.
Our first speaker in the afternoon session is Dr. Paqui Blánquez-Cano. She's a professor in the Department of Chemical, Biological, and Environmental Engineering in the University in Barcelona.
Good morning, everyone. I am going to present a preliminary study on how to implement a pilot reactor for the biological removal of pesticides from agricultural washing wastewater, which is what we want to remediate. As you know, pesticides are necessary to guarantee that there is enough food for the world population, but this means that these pesticides need to be applied following good application practices, because otherwise we can encounter derived problems from, or problems derived from the wastewater created or generated. In fact, in our agricultural sector, we generate wastewater derived from washing agricultural machinery after applying pesticides. What is normally done with this washing wastewater is to store this water in ponds or in basins until it was seen that these practices were not good, that this washing wastewater was highly contaminated, and that it had been treated before the discharge, because it would be otherwise an important problem for surface water or groundwater even. When we thought about how to bioremediate this type of water, one of the organisms that we had been using for many years were white rot fungi, WRF, which get rotten, giving out a white color, and which possess a high oxidative power because of the powerful non-specific enzyme system, both intracellularly and extracellularly. Intracellularly, it has some enzymes like cytochrome P450, but in the extracellular matrix, it has lignin peroxidase, manganese peroxidase, and lacase, which are enzymes, and they can be used for polycyclical aromatic hydrocarbons or for dyeing, etc. And we were thinking, well, we can use this fungi to be treating the pesticides that we have in our pond. And we were thinking about bacterial competition because, well, this has been mentioned here. So talking about microorganisms and having what do we have in every contaminated medium. And we had historically been working in sterile conditions in labs without problems of bacterial contamination. But we knew that if we wanted to take this process to an industrial scale, we had to remove the sterility part of the process. We cannot apply a sterile process on a contaminated medium. And therefore, we normally work with this fungus with pellets. I mean, we use the form of pellets, but it needs some external carbon source, which normally is glucose, and a nitrogen source as well. We were thinking, well, we can make trametes, which is the name of the fungi, to work on wood because this is the lignocellulosic substrate, and we can immobilize this fungi in trametes, grown trametes on splinters, wood splinters. And therefore, to compare or try to change the process, we were thinking, okay, let's compare the results from our classic bioreactor with the fungi in this shape of pellets, and we will compare it with a rotating drum bioreactor. If I compare our fluidicide bed bioreactor with a rotating drum bioreactor, working with these wood splinters, and the drum is rotating, as according to the name, and let's compare these two works with this one working with the fungus in an immobilized way in the shape of pellets. The wastewater is the same. They were coming from an artificial pond or basin in the province of Gerona in Catalonia, in the Mas Badia Foundation, which is part of the Technology and Agri-Food Research Institute. And the washing wastewater accumulated in that basin were coming from different cultures, from rice, apple, corn, and tomato crop fields. The first thing that we needed to do was to know what we had in that basin or pond, because we didn't know. The farmers were using pesticides and insecticides, but they don't really know what they are using, most of them. And therefore, we had to identify which pesticides and which insecticides, and we tested them. They underwent some confirmatory tests or analysis and quantification of pesticides. The confirmations of both bioreactors, we wanted them to be similar in some parameters. For example, the optimum pH for the fungi is 4.5, and therefore we work with this pH in both bioreactors. We carried out discontinuous treatments along 17 days. We tried the biomass to be the same, although in the fluidicide bioreactor we only have fungi, and in the rotatory one, the fungi is immobilized on the wood, but this is just fungi. The amount of glucose in the fluidized bioreactor, we had to add some glucose and the nitrogen source, and in the rotatory rotating drum bioreactor, we didn't add any nutrients. We compared sterile and non-sterile conditions as well to see the differences with our classic working method, which was sterile, and we knew that we couldn't apply sterile conditions on the field, of course. And we identified the pesticides, and we found 4 predominant ones: the thiacloprid, chlorotoluron, azothiazotocin, and tebuconazole. And we studied the parts per million, that is the concentration. When we conducted the degradation experiments here, in fact, you can see the azo example, or AZO, where we have in red the sterile fluidicide bioreactor. In green, you see the non-sterile fluidicide bioreactor, and you have the other 2 rotatory bioreactors, or rotating drum bioreactors, in the other 2 colors, in blues, the 2 blues. And we saw that we could degrade up to 100% in the first batch the contaminants. After 12 days, the second bioreactor was also, the second batch was also able to degrade. The non-sterile bioreactor could degrade the pesticide in less than 15 days. In the case of the fluidicide bioreactor, We also have that in sterile conditions, after 10 days, it is able to degrade it. But if we see non-sterile conditions, only a little bit more than 50%, that is 55% of the pesticide is degraded. And these results are repeated for the rest of the pesticides identified. As for general conclusions after these analysis, we saw that Trametes versicolor, the fungus, is involved in the degradation of the detected pesticides, because in sterile conditions, we are able to degrade approx 82%, meaning that in the bioreactor, there's only the fungus, the Biotrametes. However, in non-sterile conditions, when there is competition with other microorganisms, degradation is decreased because other microorganisms are competing for the nutrients and they do not allow Trametes to grow, the fungus to grow. And the case of the rotatory drum bioreactor, in both the first and the second batches, the elimination percentages or removal percentages are very high in sterile and non-sterile conditions, and therefore we were successful in removing the bacterial competition. But as Cosimo was saying this morning, we do not only need to know whether we have degraded the pesticides, but we need to know whether this water that we have now is as toxic as the initial one, less toxic, or even more toxic, because maybe the degradation products or the metabolites that are generated in the biodegradation process of these compounds are even more dangerous than the precursors, the precursor molecules that we had in the basin or the pond. The toxicity tests were done with microtox in the case of toxicity, and with seed growth. Seed growth, yeah, seeds. The growth of seeds, or tomato seed growth, in the case of phytotoxicity. What we were able to observe in this case was that in the drum reactor, toxicity is more reduced than in the fluidicide bioreactor, 8.6 to 6.2, comparing fluidicide with rotatory drum. In the case of phytotoxicity, we observed that the best results were obtained with the rotatory drum bioreactor compared to the fluidized bioreactor. Apart from phytotoxicity and toxicity, other parameters that we take into account when treating wastewater is the organic matter content, the COD in English. We also wanted to analyze the absorbance and the RDB. If you see both in the fluidicide bioreactor and in the rotational drum or rotatory drum bioreactor, we have an increase, which is rather acute in the case of COD. In the case of fluidized bioreactor, this increase is due to the fact that we add biofoam to avoid operational problems. And in the case of the absorbance, which also increases in the fluidized bioreactor, this is due to the fact that pellet, over the culture time, is losing part of the biomass of the pellet, and this motivates this dispersed biomass in the bioreactor. In the case of the rotatory drum bioreactor, the significant increase of organic matter is due to the extraction of soluble compounds in the matter. This increases the color as well. When we treat this wastewater, initially the color is almost transparent, and in the end, this color is turning brownish due to the extraction of the wood compounds, which makes absorbance to be increased as well. We keep under control the bacterial contamination. This was under control. After the treatment, we conducted an analysis of fungal communities in the rotatory drum bioreactor. And we saw that Trametes that was inoculated at the beginning, 100% of the fungal mass was Trameted. At the end of the process, only between 20 and 40% of Trametes were present. But other fungi were growing that had degrading capacity on these pesticides like Meierosima or Penicillium. And I mean that what we saw this morning is that populations adapt to the medium. Trametes was getting loose of the wood, but other populations were growing, were adapting to this medium, and therefore degrading the pesticides. Therefore, as general conclusions, I would say that the effluent quality was better with the rotatory drum bioreactor than with the fluidized bioreactor. We need an after treatment of those waters, maybe with activated sludges, conventional ones, to reduce the COD, and the rotatory drum bioreactor is the one offering more promising results on an industrial scale. And in fact, we have been awarded an ecological transition project 2 months ago where we are going to build a pilot bioreactor to treat the Mas Badia wastewater. Apart from the treatment in the bioreactor, we will also treat biobatteries and effluents in order to reduce the COD, and we are going to treat the wood where Trametes is immobilized in biobattery once the bioreactor is built. So thank you very much. I would like to thank the research group for the work carried out. Thank you very much. Hello, as we were saying in this session, when we mention bioremediation, we do not only speak about microorganisms, as we were told during the first part of the session, but we really refer to bioremediation using biological systems such as plants, fungi, as we heard in the previous presentation, microorganisms, and other types of cells. Protozoa. And we're going to focus on bioremediation mediated by microorganisms that uses contaminants, incorporating them into their metabolic cycle to degrade them, obtain energy, synthesize molecules, and last but not least, the synthesis and proliferation of biomass. Within the existing techniques, in bioremediation, it is possible to have different treatments and techniques, the application of which will depend on the characteristics of the medium, the nature of the contaminant, or the complexity of the contamination problem, among others. As we have said, among all these possibilities, the use of bioreactors is a viable and efficient way to tackle these bioremediation treatments in a contaminated medium. We saw a brief description of a bioreactor, but I would like to add that the bioreactor is an isolated system or environment that guarantees, where we create control conditions that guarantee and maximize the growth of a culture of microorganisms that carry out the degradation of the contaminants of interest in the aquifer. And therefore, this type of systems can provide for a continuous source of degradation that can work under controlled conditions that ensure that it will be an efficient system. The configurations and types of bioreactors are various. There are in-situ bioreactors that we install and act on the field. Then there are ex-situ bioreactor where we treat the contaminated matter independently outside, let's say, not on site. And there are other types of work of functioning or working of the bioreactors depending on the conditions that are induced on the bioreactors, and therefore we can have aerobic degradation pathways or anaerobic degradation pathways. In the case of HCH, the degradation through aerobic mechanisms is done through the LIN genes, whereas the anaerobic degradation is done through mechanisms of chlorination or chlorination mechanisms. The development or the factors that affect the bioreactor performance depend on many things, such as nature, composition of the microbial community, the nature of the contaminant, environmental factors such as pH, oxygen concentration, temperature, and even substrate or nutrient concentrations that are existing in the medium, or the ones that are giving electrons, for example. We also need to be aware of the factors related to availability of the micronutrient or the bioavailability, what we call. To implement the development of a bioremediation system of this kind, we need to, previous to the application on the field, we need to conduct some previous works such as laboratory scale works in many cases, and we need to also adapt that scale to increase the volumes that we use in the laboratory so that we can use them on the field, adding also the parameters that will affect the work on the field. Here we present the study design and development of a bioreactor for the in-situ remediation of groundwater in the Bailin aquifer. Such a process can be a middle or long-term process, and in this case it has been divided into 5 different stages. They go as following. There is an important part of laboratory work in the work and design of this working scheme. Before getting in depth into every one of the stages, I would like to say that we will base our bioreactor study on the study of aerobic degradation processes. However, we do not discard the transition to anaerobic processes that are also efficient when it comes to degrading HCH and other contaminant matter present in sites with problems similar to the Bailin aquifer. The first 2 stages constitute characterization stages where there is an initial screening of various samples that can be interesting for the following steps. In the first stage, we select a series of samples so that we can select the best candidates for the other stages. In the second step, we characterize additionally the candidates that are more successful or interesting for us. The third and fourth stages correspond to the scaling up process. In the third one, we conduct biostimulation tests and bioaugmentation tests from the candidates to inoculates that were more interesting in the previous stages. In the fourth stage, we will implement a bioreactor at lab scale where we can adjust different variables that can affect biodegradation in a more controlled way. Step number 5 is the implementation stage on the field test in one of the boreholes existing in the site. Although the flow of work that we saw was presented in a linear manner, the process in real conditions can be more similar to the workflow that you see on the slide, where we have identified every one of the stages that we saw in the previous slide, and where it can be necessary to repeat some of the experiments done because of malfunctioning of some of them, or in order to study new variables, or to optimize some of the conditions. As we were saying, the whole process involves a high number of factors that affect biodegradation and require a gradual optimization of the design process to ensure proper performance of the system. As for the location, we have selected a borehole located upwards, upwater to the Bailín aquifer with hydrogeological characteristics that we deemed interesting and ideal for our goal. This location was in an area with low contaminant concentrations that were in principle could be approached with bioremediation close to siltstones where hydraulic connectivity and the flow of underground water is lesser than in the sandstone area, where what would give us a more stable and more optimum environment for this system. We also selected an in-situ configuration because in the Bailín aquifer, the predominant type of rock is loose rock soil, and therefore we could treat directly the underground water running there. Out of the flow of work that I mentioned before, currently we have succeeded in the stages number 1 and 2, and the third one is ongoing, an ongoing process right now. We will focus right now in the goals and the outcomes. The previous stage allowed us to characterize a series of initial samples in the Bailín aquifer so that we could select the 13 indigenous samples that would be more interesting for us. There were different samples of different nature, and we wanted to compare them according to the results that came out of the analysis of different parameters. Out of the 13 samples, well, we analyzed them according to different points of view, chemical point of view, and microbiological characterization as well. And out of the obtained results, we selected 6 samples that were the most interesting according to the biomass concentrations that they contained, the diversity of microbial communities, and their tolerance to different concentrations of contaminants. In a second stage, that would consist on the additional characterization, these 6 candidates were analyzed in co-inoculation experiments so that we could verify that the metabolic activity of these samples were ideal for or suitable for the purpose. And on the other hand, we wanted to see whether the samples selected could be used as co-inoculants. In these experiments, we were able to select some samples with a metabolic activity that was suitable, that would be interesting to continue using them in the following steps of the process. We identified some mixtures of samples that improved the metabolic activity of the individual samples, and therefore they were interesting to use them as co-inoculants in the future bio-augmentation experiments. Step number 3, which is still ongoing, as I said, is being done in an orbital incubator with Erlenmeyer flasks so that we could see how the effect of every one of these effects could improve the metabolic activity of every contaminants and improve the results. And by way of conclusion, I would say that the development and the implementation of a bioreactor is a complex process on the long term due to the amount or the number of factors that are involved in the efficiency and availability of such a system. Normally, this process requires the development of a bioremediation system that is based on this principle, and it should be escalated where we control and optimize the variables related to the bioremediation processes. In the work in the Bailin aquifer, we were able to select samples that are interesting to be used as co-inoculants, and we can continue therefore with the escalating process for the implementation of a field bioreactor. Thank you very much for your attention.
Okay, our third presentation will be given by Makabat Konurbayeva. She's a microbiologist at the Kyrgyz-Turkish Manas University in Bishkek, Kyrgyzstan.
Okay. Dear guests, dear participants of the forum, let me present a presentation on application of the methods phytoremediation pesticides contaminated soils in a field experiment plot in Chimkorgon village. Excuse me my English. Okay. The wider spread of use of pesticides in agricultural practice has led to the fact that countries, in one way or another, are facing pesticide-was problems. The management and disposal of obsolete pesticides, as well as the remediation of pesticide-contaminated soils, are important global issues, important for agriculture, environment, health, and quality of life. Once the source of pesticides entering the ecosystem, all pesticides warehouse, landfills, and agriculture efficient airfields. For the Kyrgyz Republic, the problem of disposal of obsolete pesticides is relevant, since there are more than 200 storage sites in the Republic, and more than half of the unstable condition with contaminated degraded soil. These studies are implemented as part of the project Technical Assistance in Implementation of Trials on Bioremediation of POPs, contaminated soils, for successful cleaning of soil from toxic substance and application of bioremediation and phytoremediation technology in the field at the experimental site. To evaluate the effectiveness of bioremediation and phytoremediation technology for contaminated soils with obsolete pesticides, an experimental site was selected, a former airfield in the village of Chinkorgon. This site was used during agricultural works for aircraft, refueling and aerial spraying of pesticides, and was also used for pesticide storage. This place, the first chromatography analysis we done in the month of May 2021 years. You can see this date in the table. 19 types of pesticides are shown, and the amount reaches up to 10,000 milligram per kilogram of soils. This plot was divided into 3 variants. First is a variant with fertilized soil and bioproduct. Variant second is variant with fertilized soil, and variant 3 is a control without anything. Thus, in 2021, we carried out a number of activities for bioremediation of soil. The soil was without vegetation, plot of about 10,000 square meters. First, this soil was harrowed by tractor, fertilized good soil, then irrigation, and sprayed with liquid bioproduct based on the accession of soil bacterium that were isolated from pesticide burial sites and warehouse where pesticides were stored. In the table, chromatography analysis and this date can be seen in the table. comparative analysis of soils with 2021 and 2022 with, for example, endosulfan 2. For example, endosulfan in May 2000 milligram per kilogram soils, and March 0.100 milligram per kilogram. Experience variant with soil, fertilized soil plus bioproduct. And then this place, 2021, we use application aerobic bioremediation. In 2022, we use phytomediation technology on this plot. Before we wanted to use wild plants growing on the area. In the plant species that grow in this area, Harmala, Artemisia, Brassicacea, and all. But since we couldn't get these seeds, we decided to use seeds of agricultural crops. We used wheat, beet, and maize seeds. You can see the sowing scheme in this picture. So seeds for this germination were pre-treated with biological product and PGPR bacterium, growth stimulation and antifungal properties for plants. This strain is from collection in our laboratory on the basis and culture of leaf soil Streptomyces fumanus, and was isolated from soils Kyrgyzstan and represent local soil microflora. So we planted on the end of April. You can see the result and photo after 20 days after sowing. This photo shows the variant with real product for soil bioremediation was used. And this variant, experience variant with control, fertilized soils, no pre-bioproduct, experience variant with soil, fertilized soil plus bioproduct. This picture, 20 days after sowing, control variant with soil addition. This picture, next one, variant control soil with heavy pollution. As you can see, these photos, no plant grows seeds. Agreed they germinate, but cannot take root on this site. Reseeded 3 times with seed of crops, but not a single plant has taken root. This area, these variants, heavy pollutions. In the phase sprouting, plants from different variants were selected, dried, separated into above ground and underground parts and examined by chromatographic analysis. And the vase, and this picture. So in this figure, we can see that plants in the variant with grow viable, that they can grow in these doses. For example, 0.6 milligram of soils, but in the variant with heavy pollutions, the seeds can germinate but cannot grow. And near this experimental soil, livestock can graze it year-round. And we also studied the microbiological composition, the rhizosphere microflora of plants. And phase sprouting, the colony forming unit was from 5 million and 3,000 cells per gram. Especially should be noted this oligoneutrophilic group. In 2021, we are not found in this soil at all, but now we in 2022, we can note their evidence in soil variants. Also, nitrogen bacteria we are not found in these soils, but now their growth was noted. And this. Among ammonifying bacteria species, diversity was represented only by 2-3 species, mainly bacillary species, Bacillus subtilis, Bacillus cereus, and all. And the oligoneutrophil group, if early in the soil, when the contact organohalide compounds exceeded more than in 100 times, they were not detected. Their growth was completely absent. Now with the growth of plants, the quantitative ratio increased. This picture maturation phase of wheat, repining phase of beet. This picture see how in 16 months of work this piece of land has been transformed. And conclusion, when applying phytoremediation technology when growing plants, all experimental side, we can conclude the availability of this soil is established, indicate evident growth of these plants and reduction phytotoxicity of soil. And microbiological characteristic of the given soil also shown increase of microorganisms in all variants. During the experiment areas of soil where is still high concentration of pesticide, we discovered and plant growth is impossible on this soil, and seeds get germination perfectly, but they cannot sprout. And thus the experimental work lasts for 18 months. Early these soils will have without plants. The given period plants evidently grow and fructify. Thank you for organization of the forum, for the support and participate in this event. Thank you for your support in funding the project, technical assistance in implementation of trials on bioremediation of POPs contaminated soils. Thank you for attention.
Our next speaker is Carmen Lobo Bedmar. She's a researcher at IMIDRA in Madrid.
Good morning. I will be presenting this preliminary study. As we all know, soil is relevant not just for the ecosystem, but directly related to human health. Particularly in the last years, we've seen how at the European level and global level, there is a direct connection between the soil and our health. We all know that the soil is a finite resource, non-renewable, with specific functions for us, such as climate regulation and give nutrients to organisms in the soil. We also know the processes of contamination, as we all know. This person is speaking really fast. Really poses a threat for soil health. It reduces its capacity for the production of goods, and there's problems related to organic compounds. As you can see in this slide, this means so many losses and reductions. Among the pollutants, we have organochlorine, among them LINDANE, that you all very well. We know as well that there are many sites highly polluted due to the byproducts of this pesticide that was used for so many years, and we need strategies to remediate and recover these soils. We've also heard about a lot of physical chemical strategies, because sometimes the dimension, the magnitude of this contamination is so high, and the only way to carry out a recovery of these soils is using these physical chemical techniques. But if we have concentrations of pollutants that are not that high, we can also use biological techniques that are more sustainable, that allow us to work in situ and to recover these functions that I mentioned before and are so important in order to have healthy soils in the ecosystem. Bioremediation, we have been hearing about that, is how to use microorganisms or plants associated to these microorganisms in the rhizosphere to remove these pollutants or reduce the risk of their presence. We also know that soil has a capacity to buffer contamination, so its microorganisms are able to carry out a bioremediation themselves, but sometimes that is not efficient enough, and we need to improve that natural bioremediation. In this case, we have used one of the strategies to improve bioremediation. It's used an organic matter source. Among the different sources of organic matter, we used a compost derived from the treatment of wastewater treatment plants, also meeting the requirements of the circular economy, giving a new life for a residue or a waste, and this gives us a matter, organic matter and exogenous microbial population. We also need to remind that this comes from wastewater, so that means we have a very high microbial population, so we can think of a synergic or antagonic effect when in touch with the soil microbiome. So our objective was to evaluate the use of a compost from the sewage, sludge, and pruning wastes to enhance bioremediation in a soil polluted with organochlorine products. This soil is in Sabiñánigo, at the foot of the Sardas landfill. As you can see here, concentration is not too high as compared to other concentrations you are seeing these days, but we are over legal limits, and in this sort of soil, we can try a bioremediation in situ technique. The predominant contaminants were 1,2,3 fluor, 1,2 fluor of chlorobenzene and beta HCH, particularly HCH, beta HCH. Treatments tried here: natural attenuation and biostimulation using this compost, and also comparing it with a nutrient source with a mineral fertilizer and NPK in order to see whether the soluble nutrients could enhance the stimulation of the degrader microorganisms. Here you can see how we worked with this soil from the foot of the landfill and with the corresponding security measures in order to prepare and carry out the tests. Over 60 days under controlled conditions of temperature and humidity in order to see if we could could somebody please tell this lady to slow down? We were measuring the enzymatic activities in order to see the impact on the quality of the soil. We wanted to analyze enzymatic activity and see if after treatment phytotoxicity had been reduced. Let's see the results. Here we can see in this chart the degradation rate of the main three compounds that we mentioned before. We see how higher degradation was in the case of 1,2,4 and beta HCH, and for alpha HCH was rather low biodegradation. As you can see here, The compost is the gray bar, has a very positive impact on degradation. In the case of 1,2-fluor, trichlorine, benzene, differences are not that significant, probably due to the high variability of the soil. But in the case of beta HCH, we see how the impact of count stimulates degradation, and we were able to reduce the percentage. Something I wanted to highlight is that normally literature talks about how beta is the highest, or the most resistant and the most difficult to degrade. But here, compost had a very positive impact on beta more than on alpha. If we want to see this reduce in degradation is linked to an improvement in the quality of the soil, we wanted to see the impact on the enzymatic activities here. We can see how in the 6 analyzed activities, the treatment with compost, BCP, that's the right side bar, is always higher than the rest of the treatments. Why? CP, so the compost, includes organic matter and nutrients, is stimulating the microbiota of the soil, and this promotes this biostimulation. But we also need to keep in mind there are also microorganisms that can be degrading. And also, because we are applying this organic matter that is so important, we can also reduce pollutant availability. So soil microorganisms can have a higher rate of activity. So these can make us think that the application of compost was effective. However, as you see, NPK, which are soluble nutrients applied directly to the soil, have no effect at all, no positive effect, and the results are exactly the same as when we don't apply it. Because we also wanted to see potential toxicity reduction, we carried out this zucchini test in order to study germination of the seeds. As you can see here, treatment with compost, that's the right-hand bar, and index of germination increased significantly as compared to the other treatments. If you see up here, there in the zucchini scale, we can see how moderate is under 50, and under 80. And if you see with the natural and NPK treatments are really highly toxic, but when we apply compost, we are really moderating toxicity. We are recovering these functions that I mentioned at the beginning. Before reading the conclusions of this study, I just wanted to explain our method. We see this organic matter source. It was sufficient, and it looks as if really improves degradation. We are using other organic matter sources like biochar. Biochar is a residual or a waste matter coming from olive pruning. As you can see here, biochar didn't give us really the same results as the compost. It's still better. I mean, compost application is still better than biochar. We also have used tests or assays at longer terms, like 90 and 120 days, which increases degradation, and the results will very probably show a higher degradation rates. This idea of carrying out this assay is connected to a project where we are working with IMGRISA in the landfill, and we are trying a new technology of remediation to be applied on the soils at the foot of the landfill. We selected different species, as you can see here, from trees and bushes, and the treatments applied in the soil coming from the line are mycorrhizas and stimulation with this compost and the nutrients. That's why we were so interested in seeing before the growing of the plants what the effect was on the soil. Obviously, when we remove the plants in September, we will see the effect on the soil directly and on the rhizosphere of the plant itself. Sorry. And when we look into the evolution of the plants, we are expecting better results from the compost implementation. And just to read the conclusions, you see how the organochlorine compounds detected in the soil underwent a degradation process. Under the evaluated conditions, that the natural attenuation achieves degradations of around 45%, not observing any positive effect when applying soluble nutrients. However, the application of compost induces an increase in degradation to values around 57% in 60 days, probably due to this double effect I mentioned before of biostimulation due to the contribution of nutrients and organic matter, and a potential bio increase due to the incorporation of exogenous microorganisms. The application of compost shows a positive effect in the recovery of the soil functionality, because we observed increases in the biological activity of the soil, as well as the decrease of toxicity. Thank you so much.
The next speaker is Alan Sich. Alan holds a PhD from Guelph University, and his research focused, guess what, in the remediation of pesticides, contaminated soil.
Okay, what I'm going to speak to you about today is at the other end of the spectrum of bioremediation, because we're not identifying any particular bacteria. We're not characterizing them, we're not counting them, we're not doing anything with bacteria, we're not inoculating. We're using the native soil microbial population to ferment organic carbon in the presence of powdered elemental iron, and in this way create very strong reducing conditions under which the thermodynamic conditions for chemical dechlorination become favorable. This approach has been practiced at full scale since the first application in 1998, so it's been done for a long time. I'll show you some photos of commercial applications just to give you a feeling for what it looks like. I'll talk to you about the reagent that Evonik manufactures to do this. Talk a little bit about the background, how we came almost fortunately upon this formulation. Talk about the chemistry and biochemistry involved to get good pesticide degradation, including HCH compounds, and then show you 3 very brief, what I call project snapshots, not really case studies. Okay, so how is it done? Here's a typical application at an industrial pesticide site in Canada, where we're mixing the Deramen soil additive, the iron carbon additive into the soil using agricultural equipment. Here's a larger project also in Canada. This one was about 17 hectares of surface area contaminated with pesticides from strawberry farming. This is one in the state of Washington at a chemical production site where pesticides were manufactured. This soil has been excavated and put in a polyethylene line treatment cell where the additive is blended into it and water is added. This one is at, this is not even pesticides, this is at the United States Army ammunition plant in Iowa, because the same approach works very well on organic explosive compounds. And then this one is mixing of sediment from surface to 2.5 meters below ground surface in a wetland in Virginia at a United States Navy station. And then this one is at a different US Army site. This is excavated soil in what they call windrows, stockpiles, where a compost turning machine is used to do the soil mixing and addition of the soil amendment and water. The reagent that we're talking about, it has a trade name, Deramend. It is formulated using processed plant fiber, which is why we believe it's a very sustainable reagent. The plant fiber we use is a side stream from wheat processing. It's added to that is reduced iron powder or elemental iron or zero valent iron as our industry likes to refer to it. And we add a food grade emulsifying agent. Typical dosages in pesticide contaminated soil are between 0.5% by weight up to about 4%, depending on the nature of the soil and how contaminated it is. And that results in prices or costs, treatment costs between about $15 per ton of soil and $90 per ton of soil for the reagent. Then, of course, you have other labor and other costs. Good uniform soil mixing is important. Water content has to be adjusted properly. The soil has to be wet enough for this to work. And it's usually applied to surface soil, but we have done some soil mixing jobs to depths as great as 6 meters below ground surface. This is the very first observation. This was done in the Environment Canada laboratory in Canada, where I was doing my doctoral research. And what we found was that if you supplemented the soil with organic carbon as compared to a control, and this is Endrin, so it's an organochlorine pesticide. We had a stable control over a period of about 100 days. If you added organic carbon, biodegradable, fermentable organic carbon and water, you had some reduction. If you added powdered iron alone, you had some reduction, but the combination of the two, there's obviously a really strong synergy here. Something was going on, because the performance here was much better than the additive result of this and this. So there was more going on than just adding the two together. Some type of synergy was involved. And given the short time of the talk, I tried to kind of concentrate our understanding of what's going on into a single slide. So you'll have to bear with me on this. I hope I don't go too long. When you add iron into groundwater or into a wet soil, that iron begins to corrode or oxidize. At the surface of that iron particle, because of the reaction that generates hydroxyl ions, the pH goes up. The pH goes up really high, goes to about 10, sometimes even higher than 10. What happens then is that you get precipitates. Iron carbonates form, a variety of other precipitates will form, and if enough of those precipitates form, they'll coat the surface of the iron, and the iron is no longer reactive. It becomes passive, so it no longer is a good reducing agent. The way to remove that precipitate or prevent that precipitate is to prevent the pH of the aqueous solution at the solid aqueous interface from going into that range of 10. And the way we found, almost by, serendipitously, was that by putting solid organic carbon there, as it fermented, it would produce volatile fatty acids, and it doesn't matter which bacteria you've got, as long as you've got a healthy heterotrophic microbial population, they ferment organic carbon under anaerobic conditions. They produce acetic, butyric, propionic, lactic acid, all of which have pKs around 5, so they want the pH to go down. If you get the ratio right between the carbon and the iron, you can produce enough acidity here to offset the alkalinity from the iron. That keeps the surface of the iron clean, very reactive. Lots of ferrous iron comes off of it. Oxygen is consumed, sulfate is consumed, nitrate is consumed. The redox potential will drop from plus 100 to minus 500 in a matter of about 24 hours. And now you have thermodynamic conditions that are really, really good for reductive dechlorination. So that's what we're doing with this iron carbon soil additive. How does it work on HCH compounds? Pretty well. Here's some laboratory testing. Alpha, beta, gamma, lindane, total HCH compounds. You can see they aren't all removed at the same rate. They aren't all removed to the same extent because there's quite large differences in the acute toxicity of each of those isomers. There's also significant difference in their solubility, and I believe the rate of removal is most closely tied to their differences in solubility. The difference in the two Derrimend reagents was how rich, speaking in nutrient composition, the organic part is, how much nitrogen and phosphorus is in the different formulations. And then here's one more bench scale laboratory result with higher lindane concentrations, again showing the removal, 99% on lindane, 93% total. And now three very brief project snapshots. They're not really case studies. If you want more detail, I have detailed case studies. I also put a paper into the proceedings, so there's more detail there. Here's the first one. This is in California, right near the border with Mexico. Pesticide distribution site where you had pesticides and elemental sulfur spilled on the soil. What they were doing at that site is they were dipping cattle in a bath of toxaphene to keep the insects off the cattle. Kind of amazing when you think about it, that you were contaminating the cattle and then having a nice beef steak. Here's the irrigation, agricultural equipment blending the Derrimend into the soil. And here's the performance data on toxaphene and DDT. At this site, the total addition of the soil amendment was only 1%. And you can see here its note its log scale. So very good removal of toxaphene. These numbers, CHH, that's California Human Health Screening levels. This is the one for industrial for toxaphene. This is the one for residential land use. So we achieved the industrial standards for all the different sampling areas at that site. That was about 1.5 hectares of soil. And then here you have DDT concentrations, and again, log scale showing the removal after one application of the soil additive with mixing and irrigation to get the water content up. Second project snapshot, this is lindane and HCH compounds, industrial site in South Carolina. And actually it just occurred to me today, this is a bit of an anniversary for me because this project was done in 1998, believe it or not. So, and we started in May of 1998. So the 25-year anniversary of this project is coming up. It's kind of shocking when you think about it. I forget how old I'm getting. And here's the performance data from one of the sampling zones. Again, because of how much time we have here, I just tried to pick some representative data. So here you can see alpha, beta, gamma, lindane, delta, and total HCH. Oh, what am I doing here? And you can see the removal efficiency pretty good on all the different HCH compounds on this project that we did at a pesticide distribution site in South Carolina. And then the last one, this one is in the Midwest United States. I can't say anything about this because it was a lindane production facility and the site owner is very sensitive. All I can show you is the data. It looks like a long treatment time, 371 days, but you need to keep in mind that this ran through the winter in the United States Midwest. So about 120 days of that, the soil was frozen. So it wasn't really bioremediation for a lot of it. But also And also this one's interesting because it allows you to see how high, this is parts per million, not parts per billion. So 1,600 ppm of lindane, we were able to knock that down to about 133. The treatment stopped here, even though it surely would have gone lower, because they were doing a change in land use and they became impatient. They dug up all this soil and took it to a landfill after we had done all that soil remediation. So it was kind of, I thought it was wrong, but they said they didn't have any more time. All right, that is it. I don't know how bad I am on time, but anyway, this approach with the soil additive, it is bioremediation, but it's really kind of a chemical reduction approach. It's microbially enhanced carbon, actually it's using non-specific microbial carbon fermentation to make iron particles more reactive and get them to produce more reducing equivalents and sort of flood the aqueous phase of a wet soil with reducing equivalents that then do chemical dechlorination of the HCH compounds. And it has also been used on virtually the whole range of commercial organic explosives, many other pesticides and herbicides, and also chlorinated VOCs. All right.
The next speaker is Dr. Juan Pedro Martín Sanz, and he is working as a postdoctoral researcher at the University of Santiago de Compostela.
Thank you very much for the introduction. I will talk about the preliminary study of the remediation capacity of horse amendment in soils contaminated with HCHs. First of all, thank you to the organizers for this opportunity and all the partners that have made this research possible. I would like to say that the use of pesticides, particularly since the Second World War, led to an increase in crop yields, for an increase in population, but from then on, one of the most popular pesticides was the HCH, and then from the 1950s also the gamma isomer. Since 2001, alpha, beta, and gamma isomers were considered POPs, so their use was constrained or limited. We need to remember not just the impact on the soil, but also the large amount of waste generated in the process of their production. In many cases, because in those cases they were not in place, the environmental controls, their management was sometimes negligent. So let's have a look at the bioremediation as an effective decontamination technique and more eco-friendly than other techniques that could be used. In our research, we try to apply an initial evaluation of the capacity of bioremediation of organic horse amendments of soils. The area of study was in the Bilin landfill, where we took a soil sample, as said before, about the geology characteristics. We must only add that the predominant soils tend to have small development and how these the presence is more in the calcisols and others. We established 4 treatments. The soil sample from the landfill, we called it low contamination LC soil with 0.9 97 ppms. And from granulates of technical HCH in the field, we added it to another sample. We called it high contamination soil of 130 ppms. The use of horse amendment, we used a 5% for both contamination concentrations. After this addition, there was a process of 1 month of hydration and re-humidity, or in order to have a look at the presence. And then we characterized pH of the total phosphate and soluble ammonium. And then there was a follow-up with analytics for day 1, day 0, day 1, day 7, and 55 days, using the different biochemical processes, the carbon cycle with the beta-glucosidase activity, giving us information on the last part of this last cycle, and nitrogen cycle, the phenol oxidase activity, and the nitrogen cycle would be represented here by the arylamidase activity, giving us information about other properties. And also we analyze the urease activity, giving us information on the urea. We also analyze phosphatase activity, and we also analyze the dehydrogenase activity for information on the amount of microorganisms living in any of the 4 treatments. Also, we determined concentrations of the different isomers of HCH. About the results of the soil parameters, we can see how the horse amendment generated these pHs here. This is the content of total organic carbon, TOC, with an increase of between 0.93. Sorry, you can read the results on the charts, and here you see the soluble ammonium between 2.5 and 4.2 times. As you can see, horse amendment increased the amount of nutrients as expected. As for enzymatic activity, we saw how the solid red lines represent the activities in soils with high concentrations of technical HCH, and how in all the cases the activity and the enzymatic activity was the smallest. You see the dot lines represent horse amendment treated soils, which is always better. Let's see this dehydrogenase activity, informing about living microorganisms, is increasing over time, both for high C and low C. As for the phosphatase, in low contamination soils with the amendment addition, we saw an increase and then a decrease. We think this is due to the fact that part of the available phosphate in the horse amendment is then used by microorganisms, or maybe due to physical chemical processes is then not available. And then I would like to highlight that in the case of phenol oxidase, in the case of high contamination soil and horse amendment, the activities of the enzymes as compared to the soil of low contamination are quite similar. We think this is indicating that the community of microorganisms is really degrading lindane present in these soils. As for content of isomers, the different isomers, we see the soils with high contamination rates, and on the right side, low contaminations. In every case, dot lines represent a soil with a horse amendment. In all these cases, we see less concentrations of each of the isomers of technique technique HCH. Here we can see how, while in alpha we have an important decrease that is kept over time, beta isomer degradation is not that significant. We think it is due to the recalcitrance activity or characteristic of this isomer, as pointed before. Also, I wanted to highlight that in many cases we're not really reaching an asymptote in the concentration of lindane isomers. We think that over time we could see higher degradation rates. As for low contamination soils, we also face the problem or the issue that many concentrations of the isomers are below 0.5 ppm, which was the value in the measure. So the results must be taken with caution. Anyway, we always watched a decrease in concentrations. So as conclusions, we could say that organic horse amendment increased nutrient availability in soils. Also, it stimulated soil enzyme activities, and that this use of this horse amendment could be effective in the degradation of HCH isomers, and that this reduction did not reach an asymptote at the end of the experiment. So over time, with longer time frames, we could reach better degradation rates. Thank you very much for your attention.
My question is for Alan. Very nice presentation. Most of the cases you presented correspond to soil. Can you apply this technique, and you have experience, for groundwater remediation?
We have one project for groundwater that had lindane. It was at a site in Alabama, and the same additive was injected under pressure, and they monitored lindane over a period of about 9 months after the injection, and I can send you that data. So the answer is yes, it appears to work. I think the numbers were something like 200 ppb, 200 micrograms per liter were there at the beginning, and the reduction was about 75% after 9 months. But I'll send you the data. You can look at it yourself. You're a groundwater guy, and it's groundwater data.
Thank you, Aurora Santos from Universidad Complutense. Thank you for the nice presentation to all. Very, very interesting. For me, I'm not an expert in bioremediation, so maybe I asking not very technical questions. Is for Alan the question: when you use carbon with iron particles, you know if there is a absorption on the activated carbon? I mean, the removal is explained by reduction, chemical reduction, or also absorption contributes to this removal?
Well, I think it's important to make it clear that it's not activated carbon. It's not the type of carbon that you use in activated carbon filters. It's processed plant material that has been heat treated and ground down to a particular particle size. So it's a mixture of lignin, cellulose, hemicellulose, amino acids, and there is some transient adsorption of organics onto the surface of that type of plant matter. And we know that because we've done radioisotope mass balances on it. But if the question you're asking is whether the removal is a result of adsorption fooling the analytical, the answer is definitely no, because 18 hours of boiling hexane acetone in a SOxLit or a SOxtherm will take pesticides off of any kind of organic material. I don't know if I answered the question.
I have a question for Alan, actually. For the open field application that you showed, how do you apply the product in order to guarantee homogeneous spreading? And did you observe any effect from external factors such as rain, for example?
Well, we're just using the same type of agricultural machinery as is used for spreading fertilizer onto the surface of soil. And then we have some specialized soil mixing equipment that is manufactured in Germany that allows you to very thoroughly mix soil to a depth of 60 centimeters in a single pass. So I would say that we're not getting perfect distribution of the reagent through the soil in how we spread it onto the surface of the soil, but then with 2 or 3 passes of the mixing machinery, we get very good distribution of the amendment through the soil. And then adding water is also done with large-scale agricultural irrigation equipment. So I guess the point of all of that, to make the point out of all of that, is it's not that critical to have perfect distribution, because the destruction of the pesticide is not at the surface of the iron particles. I sort of tried to talk about that. I really believe it's the ferrous iron that comes off the zero-valent iron particles that is the reducing equivalent that's dechlorinating the pesticide. I think it's ferrous iron, and of course ferrous iron is fully soluble in water, and the concentration will equilibrate through as long as you have the water content high enough that you have a continuous network of water-filled pores, so the reducing equivalents will distribute throughout the soil matrix.
So depending on the type of soil, you might need to irrigate in one way or another, depending on how much you want to saturate it, let's say.
Oh yes, and it's more difficult to treat soils that are very high in clay content, because then the diffusion of your ferrous iron and your organic carbon, the fermentation products, through a sort of aggregated clay soil is very slow. So typically the clay soils take longer than a silty soil, and a silty soil takes longer to treat than a sandy soil.
Thank you. Question for Juan Pedro. Why did you use horse manure? Did you actually compare it to other, like cow manure, for example, before choosing a horse? Was the manure stabilized? And have you investigated the impact of such manure on local microbial community, autochthonous microbial community?
Yes. Well, we had the option to have access. We have the option to have access to commercial amendment or manure or whatever. And yeah, in this case, we were also considering antibiotics, and therefore it was easy to have access to a commercially distributed amendment or manure. Yeah.
Okay, I again enjoy a lot all the presentations. I have a question for the guy, our fellow from ICOM, okay? I like your presentation. And you went through from the bioreactor to the in-situ. Okay, my question is about the in-situ test. If I understood, it wasn't clear. You plan to do that in the sandstone or in the limolites? And then the question is, okay, in the lab you control the conditions, but in the field is the host rock. Okay, how do you plan the test to make sure you will see something? Okay, is it advection? Is diffusion? What do you? Okay, how are you going to manage the conditions in the rock? Okay,
we have to figure it out. We are not sure at the moment how to monitor all the parameters at the field, but we believe that in the siltstones the main principle it will be diffusion because the advection flow is not really high because the connectivity is much lower than in the sandstone. So we will try first to adjust the parameters at the lab scale, but as you said, it's really different from reality in the field. So yeah, and this is why we choose this borehole, because it will be a more stable environment for a bioreactor. And also the concentrations in groundwater are much lower than in sandstones and are affordable for the bioremediation. It's impossible to bioremediate the sandstone at the moment in the Bailin landfill.
Okay, now the point, we have worked on that type of experiments, but in granitic rocks. Diffusion will be so slow that, of course, it's more quiet, but you will reach steady conditions because there will be nothing changing there. Diffusion is so slow. So, okay, maybe we can talk about that later.
Yeah, I'm not a hydrogeologist, so I'm open to any advice.
Because, of course, you get closer to reality, to the rock, but you will see very little things, very small, because nothing is changing there. Yeah. Other than what you put in your borehole, right?
Yeah. Also keep in mind that we are going to start working in an area upstream the source. Okay, so we will try first in this area because the concentrations are much lower than in the source and also than in sandstone. So yeah, okay. We will try to try this technology in this borehole first.
Okay.
I just have a general question or comment for everyone who's working in the area. Do you always take into consideration that on longer term experiments and depending on the temperature, that some of the changes that we're seeing may be influenced by volatilization? Because certainly these HCH compounds are a lot more volatile than things like DDT or dieldrin or chlordane. Just really wondering, has anybody sort of quantified that? Yesterday, someone was showing their data where they were capturing the volatiles on a polyurethane foam trap, and we've done that in some of our work. I'm just wondering if any of you folks have.
Yeah, I wanted to ask a question related to the compost. Could you please confirm that the sludges that are used are stabilized in this compost? Because one of the sludge problems is the amount of pathogens that they have. And I know that over time, the soil is able to remove or eliminate those pathogens, especially in very fertile soils with a lot of organic matter. But the problem can appear in areas close to aquifers where water can, well, those pathogens can go and move towards the water and go and be present in the water. Yes, the sludge that we use is composted with the rest of clippings, of wood clippings. If the compost proportion is all right. There should be no pathogens, but as we're talking about large batteries and large amount of product, in some cases we do have the presence of pathogens, but as it is applied to the soil very rapidly, normally they disappear with temperature. Yes, I wanted to contribute something to your question regarding whether we see influence of temperature or by temperature. We can say that in terms of bioremediation, thanks to the studies by the colleagues from Barcelona, the temperature that we have in the Sardas aquifer is kept at a rather constant level all throughout the year. In that specific case, the bioremediation that could take place there through biostimulation would not be altered or influenced because there are no great variations in temperature. I wanted to ask you whether you performed some previous treatment on the baleen soil before mixing it up with the compost. I'm saying this because of the treatment achieved. This is very interesting for me. Or with beta, I mean, treated with beta. Yeah, the Sardas, the other site in Sabiñánigo. No, the treatment was done, only incubated in a chamber with humidity, temperature, and aeration, and we applied the compost. It is true that beta can present more problems, but it has worked better than alpha. And therefore, we are extending the times right now. So there was no previous filtering or scanning. Well, you need to really filter the soil when you take it. If you saw the photographs, it was filtered at 5 millimeters, sieved, right? It was sieved, better than filtered, sieved.
I think we can call it a day then. Thanks to everyone.